Just trying to figure out where to put this. I think it will disappear once I get started. Okay. 40 more seconds. Okay, so I think we'll get started now. So let me know at any time if my screen cuts off, if my audio cuts off, or if everything just disappears. So essentially, I'll be starting off with biochem, and then later on in the next lecture, Akhil will be doing metabolism, and they're very closely linked. So I just had to show this off because I found it the other day and I thought it would be fun. So biochem looks really scary, and you do have to know most of the stuff. That wait, sorry, I just got a text from Akhil. I'm not sure if it's relevant. Oh, okay, don't worry. So essentially biochem is quite a high yield topic. So in my lecture, I'll be talking mainly about like the enzymes and the acid base, body fluids, and macromolecules, and that will lead nicely onto Akhil's topic, which is the actual like scary metabolism stuff. So I'll probably be spending a little bit more time on osmolarity and tonicity and the acid base stuff, because that's what most people have trouble with at this point in time. But if you want me to cover some more stuff, just type it in the chat what you're sort of struggling with, and I'll see if I can squish that in. And as always, you can always just message me if you want some help individually. So starting off with fluid distribution, the stuff that's highlighted is like stuff that's the most important on the page. So you should know that in the body, we are 60% water, and 2/3 of that is intracellular, 1/3 is extracellular. So a lot of people get this mixed up. So you just want to imagine having cells in a tub. Do you expect to see more cells, or do you expect to see more of the extracellular fluid? So this looks more like what we would expect from a setup. So 1/3 on the outside and 2/3 of it is actually in the middle. All of the other stuff, you don't really need to know. It's not like super high yield. They usually just test this and this. So osmolarity and tonicity. So it's really important to understand the differences. So osmolarity is the amount of solutes in a solvent. It doesn't matter what the solutes are. It doesn't matter if they're charged or not. It's just the number of things within the solvent, which is usually water. Tonicity is a little bit different. So this is only taking into account the effect of osmoles, which means things that actually cannot pass through the plasma membrane, and they're called impermeable or effective osmoles. So just keep in mind that tonicity tells you the end effect of the solution on a cell, and it's relative. So you can't really say that this solution is hypertonic. You can only say this solution is hypertonic to another solution. So it's all relative. So tonicity doesn't usually have units. It's only a comparison, and it tells you what happens at the end when you have equilibrium again. It doesn't really tell you what happens throughout the way to reach equilibrium. So again, terminology. So you can have hyper, which means too much. Hypo means too little. Iso means just enough. So it's like Goldilocks. They're not the same, but they can be related. So hyperosmolar, hyperosmolar, isoosmolar, it doesn't really tell you too much about what happens to the end after equilibrium has been really established. Hypertonic tells you that the solution on the outside of the cell has hypertonicity, which means it has more effective osmoles, which means that water is going to flow into the cell. Outside of the cell, and that will cause the cell to shrivel. Hypertonic means that there is more water, or you can say less solutes on the outside of the cell that can't dissolve in. So water will flow from outside to inside of the cell. Okay, So this is diffusion, just to recap from if anyone didn't do biology. So diffusion can happen in many different ways. We sort of assume in this topic that we're only talking about passive diffusion, and we're sort of assuming that it's just red blood cells. But of course, if they tell you specifically we're using a cell with like 400 millimoles that has sodium pumps, then you would change it a little bit. But essentially, we just assume that we don't really have any active transport channels. So if there's sodium, sodium doesn't actually go in, because sodium is a charged molecule that needs either a specific carrier or active transport to bring it into a cell. So we're sort of just assuming that if you can't go in with normally, then you're going to stay out of the cell and you'll be an effectively impermeable substance. So good to memorize some of these, especially, well, only the ones that you will need to know is urea. So urea is freely permeable. Glucose is freely permeable, but through a channel. It doesn't really matter with this topic, but just know that it's through a channel because it's slightly larger. And also know that glucose will slowly go in compared to urea. And water is always freely permeable, and all your charged things, they require specific channels to go through. So we're going to assume that the membrane doesn't have any of those, so they're all going to be effectively impermeable and contribute to tonicity. So this is sort of what we're going to be thinking about whenever we're trying to figure out what happens to a cell when you put it in a solution. So the first thing that you're going to try to do is move the solutes. So this means if there's a difference between the concentrations, you want to see if any of the solutes can move in or move out to reach equilibrium again. Usually we would assume that the stuff inside the cell can't move out. So it's always talking about what the cell is in, the solution that the cell has been put in. If that doesn't happen, so if you have effectively impermeable solutes in the extracellular solution, then we're going to try and move water. So first thing to think about is whether or not the solutes can move to balance the concentrations. If that can't happen, then we're going to be moving water. So if you think about the equation for concentration, that's moles over volume. So first thing, we're going to try and change the moles, and then we're going to try and change volume, and that will have, the same effect on concentration. So definition of osmosis. So this is another thing that is important to know, but they probably won't really test you on it. So they won't ask you, can you write out the definition of osmosis for 1 mark, but they'll expect you to know it. So osmosis is the passive movement of water from low solute concentration to high solute concentration. So again, with the equation C equals N over V, what we're trying to do is, if there's more moles on this side, we're going to try and increase the volume on this side to reach the same concentration as another solution that has fewer moles. So this is what you would see. And osmosis is influenced only by tonicity. So saying something is like hyperosmolar doesn't actually tell you whether or not osmosis will happen. Because if I say that this red blood cell has been put inside a solution that is hyperosmolar, all I'm saying is that there are more particles on the outside of the red blood cell. I don't actually tell you whether or not those particles or those solutes can actually move in and out of the cell. So if they are all diffusible, then water won't actually get a chance to actually move, because remember, we talk about, I don't know how to go back, but we think about whether or not the solutes can move, and then we think about whether or not water can move. Oops, I think it was stuck for a bit. Okay, so osmotic pressure. So you probably saw the lecturer draw a couple of these on his paper. So this is the pressure needed to stop osmosis. So if you think about, let's say this little U-tube, you have a solution that has solutes in it. You have a pure solvent, which is a fancy way of saying water. It's separated by a semi-permeable membrane. So under these circumstances, you think about whether or not the solute can move. Usually we'd assume that for this case, the solute can't move. So water will move into the green space to try and balance out the concentrations. And then osmotic pressure is the pressure that you have to push down on in order to stop the water from moving. So it's sort of just the pressure needed to stop osmosis. All you have to really memorize is it is affected by concentration and temperature. So concentration will affect osmotic pressure. Temperature will also affect osmotic pressure. And that's using this equation, which you probably have seen very briefly in a lecture. Oops. So don't really have to worry too much about memorizing this because I'll give it to you, but just understand that because your osmotic pressure is sort of proportional to temperature and also proportional to molarity of the solution, then it's proportional to increase in concentration and increase in temperature. Okay. So a little bit about red blood cells. So please memorize that their tonicity is 300 milliosmoles per litre or millimoles per litre. It means the same thing in this case. So 300 is the number to memorize. If you put it in something that's hypotonic, so remember, tonicity tells us what happens at the end of it, whereas osmolarity doesn't. So if you put it into something that's hypotonic, we know that hypotonic means there's less solutes, less diffusible solutes, less non-diffusible solutes on the outside of the cell. So what happens is, there's less. So what happens is that water is going to go from the outside of the cell to the inside of the cell. And the way that you can memorize the effect of it is hypo has an O and pop has an O. So you can say that if you put it into a hypotonic solution, you're going to pop the cell. Isotonic is like very nice. Nothing happens. There is no net water movement, but just keep in mind that osmosis is happening all the time. So even though there's no net movement, you're still going to have water moving in and out just at the same rate. And just a little bit about hypertonic solutions. We don't really see this in questions or in practice. So we're only usually thinking about injecting urea or saline or dextrose, whereas not really thinking about hypertonic solutions. And why this matters. So you want to assume that because your red blood cell is a tiny, tiny little volume and it's within like the tissue, which is a really, really large volume compared to the red blood cell, that basically means that the extracellular fluid is basically infinite and there's very little that will change about the extracellular fluid. So a difference in tonicity will cause a large change because the concentrations will never equalize. So we'll go into this in a little bit more detail later. First thing is normal saline or 0.9% saline. Just be careful that you don't pick one that says 9% saline, because if you just memorize a 9 but you forget that it's 0.9, then you might be in a little bit of trouble. So 0.9% saline is isoosmolar and isotonic to red blood cells. Isoosmolar means that they both have the same number of things within the liquid. And isotonic means that they have the same number of non-diffusible things within their solutions. So sodium is effectively impermeable. So technically it actually goes into the cell and then is immediately pumped out again by some ion channels that you don't have to know about yet. So therefore we just assume that it can't go into the cell. So remember, because the solutes can't go in, then we think about the water. So if the saline can't go in, then the water might go out or it might go in, depending on what happens. But in this case, because it's 0.9% saline, the water from the saline will enter the extracellular fluid and stay there. Okay, so that just means that the water, once you inject it, won't move into the cells. There won't be any water that moves out of the cells either. And you would inject 0.9% saline when you want an increased extracellular fluid. So this can be used to increase blood pressure or to treat dehydration. This one, 5% dextrose, so again, memorize it's 5%, not 0.5%, not 50%. It is the same as glucose, so you can sort of use them interchangeably or think about it interchangeably. So again, this is isoosmolar and isotonic to red blood cells, which means it has a tonicity of osmolarity of 300. So when you inject dextrose into your body, it will initially increase the extracellular fluid because it's isotonic, which means it's not going to cause water movement at the end. But eventually, because your cell is actually undergoing a very big cell respiration and using up glucose, then that causes the amount of glucose within the cells to gradually decrease. So if the outside is the same and the inside is gradually decreasing, then suddenly the outside becomes slightly hypertonic, which causes water to actually enter the cells. So eventually all your dextrose will be metabolized. So it takes quite a while, which means water won't rush in very quickly. it's going to go in very slowly, proportional to how fast your glucose is getting metabolized within the cell. So main thing to note is that dextrose will increase your total body fluid because you've got some of that fluid from the dextrose remaining outside, and eventually you're going to have some of that fluid slowly entering the cell. So it's sort of a proportional increase to both intracellular and extracellular fluid. So let's say you have like someone who's diabetic and they are dehydrated and you're not really sure whether to give them saline or dextrose. It would be better to give them saline because dextrose, remember, is glucose. So giving dextrose will actually, well, it will probably increase their blood levels. So if you've got someone who's resistant to that or shouldn't have changes in blood levels, then you would give them saline, but they have similar effects. So they can be both used for dehydration. Urea, so this one is, I'm not sure about how you guys are finding it, but I found this one a little bit challenging to understand. So first of all, memorize it's 1.8% urea because they can just change the percentages. They could give you, hypothetically, a question, all the options are urea just with different concentrations if they want to test that, but I don't think they've done it before. So it's not used clinically, which means it's damaging. So they only use it to test you. So we're never going to give a patient urea unless you want to get sued. So urea is isoosmolar. What that tells you is at the start, once you inject it, there's the same number of urea plus other things inside the 1.8% urea, and there's the same number of random particles inside the cells. But it's not isotonic. So remember, isotonicity will tell you that at the end, when equilibrium has been reached, there's no change in cell volume, no change in extracellular fluid volume, no movement of water. So it's not isotonic because throughout the process to reach equilibrium, we're actually going to have water moving and we're going to have solutes moving. So because urea is freely diffusible, it's going to flow down its concentration gradient. So in the cell, you have no urea. Urea is not found in cells. So when you're injecting urea to the extracellular fluid, what you're doing is you're introducing a high concentration outside, nothing inside the cell. So urea is going to go inside the cell. So that's why urea flows into the cell, even though it's isoosmolar. So remember, isoosmolar doesn't actually tell you the concentrations of individual substances. It's only telling you, well, if I count all these particles, I've got the same number of particles inside versus outside. But each particle type is still going to go and establish its own concentration equilibrium. So urea, even though it's isoosmolar on the outside, it's actually highly concentrated on the outside compared to inside. So that's why urea is going to go into the red blood cell. And because it's now in the red blood cell, suddenly the red blood cell, the osmolarity will increase. So that's what you have here. Okay, so adding urea to the blood, it's not going to have a big impact on the blood because 300 milliosmoles is tiny. The blood is many, many liters, so it's not actually going to cause a very big change here, but the cell is tiny. It's not even going to be a liter. It's like tiny, tiny, tiny. So adding this much into the blood, some of it going back into the cell is actually going to cause a cell to essentially double in the osmolarity. And now when we're thinking about the difference in osmolarity, that will tell us where the water is going to go. So urea is going to cause your cells to pop. It's slightly different from water. The effect is the same. It's going to cause your cells to pop. But the difference is water is not isoosmolar, whereas urea is isoosmolar, but they have the same effect. So don't pick it in a question unless it's asking you about cells popping or, that's basically the only time you would pick it. Just another little summary table for you to use for revision, just summarizing what we were talking about before. So difference between water and urea is that water is 0 osmoles and urea is 300 milliosmoles. So osmolarity is different, the effects are the same. Okay, so quick question. You guys can feel free to type out your responses in the chat. I'm not sure if I'll get a notification to see them, but I might wait like 10 seconds, get you to think about it, and then I'll go to the next slide with the answer. So you place a cell with an internal concentration of 300 milliosmoles in a 400 milliosmol solution. What happens to the volume and the osmolarity of the cell at equilibrium? I also don't know how to, oh, there we go. Okay, so hopefully everyone identified that there's actually two things that we have to think about for this question: what happens to the volume and what happens to the osmolarity, because you might have a change in osmolarity that will cause a change in volume. You can have a change in volume and a change in osmolarity. You can have no change in volume or an undetermined change in volume and an increase or a decrease in osmolarity. So volume is impacted by tonicity. Because remember, tonicity will tell you what happens to the cell or what happens to the solution over time, whereas osmolarity is just the concentrations at the end of it. So they're different things. So we actually can't figure out what happens to volume because they haven't told us what the tonicity of the solution is. Is it hypertonic or is it hypotonic to the cell? We can't assume because all they've said is that we've got 400 things. It hasn't told us that we've got 400 non-diffusible things that will contribute to tonicity. So we actually don't know what the change in volume is going to be. Let's say that everything in here is non-diffusible, such as, let's say, sodium. Sodium ions, non-diffusible to red blood cells. We're going to assume that because it's 300 milliosmoles. So because it's non-diffusible, that is going to cause the extracellular fluid to have a higher tonicity, hypertonic, which causes cell water to come out into the extracellular fluid, and then the cell will decrease in volume. But let's say that everything in here is diffusible. So if everything here is diffusible, let's say maybe it's urea. If it's urea, then some of that urea is going to go into the cell and then cause water to follow. And then you're going to have an increase in volume. So we can't actually tell what happens to volume unless they tell us what the percentage or what the concentration of the non-diffusible elements are. But we can tell what happens to osmolarity. It's sort of like an equilibrium question. So if you've got 300 milliosmoles and you've got 400 milliosmoles, eventually you're going to have to get them to be equal. So the cell needs to sort of compensate and go up a little bit. So osmolarity will increase, whereas volume could go either way depending on what the tonicity of this solution is, which they haven't told us. Next question. Which would have a greater effect on the volume of a cell at equilibrium? Solution A, which has 300 milliosmoles of salt and 200 milliosmoles of urea, or solution B, composed of 200 milliosmoles of salt and 300 milliosmoles of urea. So these questions, you probably won't see them in the exam because exams are usually multiple choice for this topic, but it's just good thinking. So it will hopefully get you to think a little bit deeper than just doing multiple choice questions. I'll give you like 15 seconds. Okay, so hopefully you've realized that what they're trying to test is your diffusible and non-diffusible substances. So NaCl, that's like injecting salt. 300 milliosmoles means that it's isoosmolar. Urea is diffusible, so it's going to go into the cell anytime that you inject it into the body. So solution A has 300 millimoles of non-penetrating solutes, which is equal to the concentration in the cell. So there's actually no gradient to cause net water movement. So the cell volume at equilibrium should not change because solution A is isotonic to the cell. Over here, solution B composed of 200 milliosmoles of NaCl and 300 milliosmoles of urea. So solution B has 200 milliosmoles compared to the cell's non-penetrating solutes, which is 300 milliosmoles. So the non-penetrating solute concentration is higher in the cell. So the cell actually has a higher amount of non-penetrating things. So water is going to move into the cell. So water is going to move into the cell, making solution B hypotonic. So even though they both have an osmolarity of 500 milliosmoles, solution B is going to have a greater effect on volume than solution A. So this is just trying to show you that an hyperosmotic solution can be isotonic or hypotonic, depending on the actual composition. So isoosmolar doesn't really tell you very much unless you're given extra information. So next one, knowing that a solution is hyperosmotic tells you that it is also hypotonic. True or false? Okay, so for this question, the answer is true. So it's a little bit contradictory to what I just said before, which I said that knowing the osmolarity doesn't really tell you anything about the tonicity. So this is basically the exception. So if you are hyperosmotic, that means you have less things in the extracellular fluid. So let's say that we're comparing the extracellular fluid to the cell. So a hyperosmotic outside solution, extracellular fluid, can't have more diffusible elements than the intracellular fluid. Oops. Just check the chat. Water moves outside of the cell. Oops, sorry. I think they were meant to be private messages. I'll look at the messages later and then I'll reply to them individually, maybe. I think that would work a little bit better. Okay, so knowing that a solution is hyperosmotic tells you that it is also hypertonic. So true or false? So this one is false because hyperosmotic only tells you that there's more particles or more solutes outside of the cell. It doesn't actually tell you if those solutes are diffusible or non-diffusible. So that's why it is false. Okay, so moving on to acid-base physiology. So for this one, you just have to memorize the numbers. Sorry, I should have highlighted that in yellow. So neutral pH at 25 degrees Celsius is 6.8. So memorize this number. This is neutral. Normal body pH is 7.4. Usually they'll just say normal pH and neutral pH. They won't tell you it's a body pH. So this is due to higher body temperature. It's also due to the fact that the body is different from water. So the composition of the body contains different ions and stuff that will all impact the pH. The acceptable range is from 7.36 to 7.44. So it seems quite small, but because pH is a logarithmic scale, a tiny, tiny increase or a tiny decrease in pH is actually really, really big in real life. So what this means is even though neutral pH is 6.8, a neutral pH is actually acidotic. It's actually, if you have a pH of this, you'll probably not have a very good outcome. So anything below 7.4 is, we'll consider acidotic. Anything above it is considered alkaline. So you can have respiratory or metabolic acidosis and alkalosis. So respiratory means that your body isn't very good at pumping out CO2 because CO2 is going to make your body acidic. So if you have defective, let's say alveoli, and it's not very good at doing the gas exchange that it's meant to do, and you're not able to get rid of carbon dioxide, then you will have respiratory acidosis. Let's say that your lungs are super, super extra good at pumping out CO2 and you actually lose too much of it, then you might have alkalosis. Metabolic acidosis and alkalosis is talking about like body buffers. So body buffers include the bicarbonate and carbon dioxide mechanism. So what they do is that they try to buffer the acids. So if you've suddenly got an increase in acids in your body, you've got these pre-made buffers that are going to go and attach to them and then try to neutralize them. So if you don't have enough of those buffers, you can get acidosis as well. And if you lose too many of the buffers, you can get acidosis because there's nothing buffering it. But you don't really have to worry about this. Just memorize the numbers for now. Memorize these highlighted things and the 7.4. Okay, so moving on. So most of the rest of the lecture is going to be revising a little bit of biology, a little bit of chemistry from what you would have done in year 12 and just adding a little bit more information to that. So cellular energetics, so this is like basically a scary way of saying the energy graphs that you've been seeing in unit 3 and 4 of chemistry. So exergonic means that energy is being released. Endergonic means that energy is being absorbed by the reaction. Free energy, you can sort of think about it as just energy, energy that is able to be used. So that's slightly different from catabolism and anabolism. They are closely related. So exergonic means that you are breaking things down, and endergonic means that you are making things, but the definitions are slightly different. So something that's catabolic means breakdown of something bigger into little things. Exergonic means energy release, free energy release, even though they are related. So hopefully you already know the structure of ATP. So ATP has 3 phosphate groups, that's the important part. And the bond, the high energy bond, is between the second and the third phosphate group. So if you break that, then you have free energy. And if you put energy into it, then you can store it up for later. This is an example of an endergonic reaction that you'll probably look at in the next hour. So this is adding phosphate to glucose to form glucose 6-phosphate. So the difference between glucose 6-phosphate and normal glucose is that glucose 6-phosphate can be used to make other things. So it's sort of like the building block to other things, whereas glucose is able to diffuse out of the cell and be used for energy. So if we want to keep glucose within a cell, then we're going to add a phosphate to it so it can't diffuse out. It's endergonic because you're making a bond, you're adding phosphate that requires energy. And so that's endergonic because you're putting energy into the bond, essentially. So as shown by the ATP becoming ADP. So try to die. Okay. So moving on. So this is a little bit about enzymes. Enzymes, biological catalysts, just know that they're made of protein. Know how to identify what an enzyme is. So if it ends in ASE, that means it's an enzyme. This isn't there are exceptions though. So there are some enzymes that won't end in ASE, which are still enzymes. So things like pepsin, that's not going to be ending in ASE. But you don't really have to know that for now. So they lower the activation energy by providing an alternate pathway. Just main thing, knowing that they lower the activation energy, therefore increase the rate of reaction. They do not impact the free energy delta G, which is the same thing as delta H. So hopefully you all know this already. They're reusable, which means that they're not changed throughout at the end of the reaction. Blah, blah, blah. Just some terminology. So the substrate, that should be substrate. The substrate is what the reactant is. So what's actually going in. There could be many substrates that will undergo the same reaction together. So you could actually have these 2 products becoming the substrates of another enzyme and then making them into one thing at the end, which is actually an anabolic reaction. This one showing a big thing being broken down into little things is a catabolic reaction. The active site is what gives the enzyme its function. So if the active site isn't working properly, or if the active site shape has been changed or denatured, the substrate can't bind. So it's very closely linked. The enzyme substrate complex is after they've binded. These are some things that can help enzymes to function. Sometimes they're required, sometimes they're just there to increase the rate of reaction. So prosthetic groups, they are permanently bound really, really tightly, and they're inorganic. So they're usually added during the production of the protein. Inorganic cofactors, they're permanently bound loosely. So things like iron, copper, zinc, calcium, for instance, iron is an inorganic cofactor that's essential for hemoglobin affinity for oxygen. So these are usually required for enzyme function. Coenzymes are not permanently bound. So what they do is that they will be there for this one reaction, and then after the reaction is over and the products are released, they might leave. They'll be changed a little bit most of the time, and then they'll be reloaded and then go to another enzyme and do the same thing. So they can add or remove chemical groups and be carriers of specific functional groups, which are usually like hydrogens and electrons and stuff. So just knowing that prosthetic groups are very tightly bound and knowing that inorganic cofactors are usually, what are these? Metal ions? Yeah, metal ions. And also knowing that coenzymes are changed throughout the reaction and not permanently bound. So they're kind of like going from one reaction to the other. That's basically what you need to know for this one. So if they ask you what sort of, what role does iron play in hemoglobin action? You would say it's an inorganic cofactor that is loosely bound and allows the hemoglobin to have high affinity for oxygen. Okay, so 2 models for enzyme function. There's lock and key, and then there's induced fit. So usually they'll ask about one of them. It doesn't always come up, but it's still something that you just should know. So lock and key model means that the active site doesn't change throughout the reaction. Induced fit model means that the active site does change throughout the reaction. So the active site, remember, is the place where the substrate will bind. If it changes, it will change to better fit the enzyme. Better fit the substrate. So induced fit, there's an active site that initially isn't completely complementary to the shape of the substrate. It is still specific though. And then after the substrate has attached, it will sort of mold itself around the substrate. So there's many different theories or pieces of evidence for each of them. For instance, for lock and key model, there are some, let's say some isotopes of substrates that will cause it not to react. So that's what has got people thinking, well, that means that the substrate has to be super specific, which means that the active site has to be super, super specific, which means it can't just mold to fit anything. Whereas for the induced fit model, they've got evidence that different isotopes can actually cause a reaction of the same enzyme. So that's, yeah, it might actually be a mixture of both. So they're not entirely sure just yet. Enzyme inhibition. So this is actually relatively high yield compared to most of the other stuff that I've been talking about. Most of the other stuff with enzymes is background knowledge, whereas enzyme inhibition, it's sort of background knowledge, but they will like to test it. So competitive inhibitor, it competes with the substrate and will bind to the active site. Uncompetitive inhibitor is going to bind to the enzyme substrate complex, not on the active site, and prevent release of products. So if you've done 3-4 biology or year 12 biology, you probably were familiar with competitive inhibitor, non-competitive, and negative feedback, but probably haven't seen the uncompetitive inhibitor before. So uncompetitive still means that it's not going to compete with the substrate, it's not going to bind to the active site, but it prevents the release of products. It's not going to prevent binding, but it will prevent products from being released. So they have the same effect. They're both going to inhibit it, but in different ways. Non-competitive means that it's not going to compete with the substrate. It's going to bind to an allosteric site, which is somewhere else on the enzyme that will change the shape of the active site. Okay, so that will also cause a substrate to no longer be specific. Negative feedback is what happens in basically all biochemical pathways in your body. So the end product or one of the intermediates is going to go back and act as an inhibitor for one of the enzymes in this chain. So sometimes it's like the first enzyme, it could be any other enzyme. For instance, if B goes off and makes another molecule as well, then you might not want to block the production of B because then you'll block the other pathway as well. So negative feedback is usually self-regulatory. And it's very important to know because they could give you a diagram and ask you what's going on. Over here, so this graph just showing you the difference between, or practical difference between a competitive and a non-competitive inhibitor. So if you're competitive, you're binding to the active site, so you're fighting with the substrate. If you increase the substrate concentration, then the substrate can actually knock the competitor off. Whereas if it's non-competitive, you're not actually fighting with the substrate for the active site. If it's uncompetitive, same thing, you're not fighting for the active site. So increase in substrate concentration won't actually have any impact on the rate of reaction after the inhibitor has already been bound. Effective temperature on enzyme function. So high temperature is going to irreversibly denature the enzyme by breaking hydrogen bonds. Low temperature is going to reversibly decrease the rate of reaction. Okay, so for human enzymes, because in medicine, we're talking about humans. So humans, will have an optimal temperature of close to 37 degrees Celsius. If you go too much above that, you're going to have a really rapid decrease in rate of reaction because your enzymes are going to be denatured, and all your other proteins as well. pH, similar, so it's going to break the bonds, but not really just break them. They're going to break them and reform them. So it's going to change the ionic interactions between amino acids. So hopefully everyone's been exposed to this in chemistry before in year 12. Just note that one of the other differences between temperature and pH in terms of human enzymes is that temperature is usually the same for all enzymes in humans, whereas pH can vary quite widely. So different areas in the body will have different pH, therefore the enzymes in that region of the body will have a different optimal pH. And also just recognizing that the way that you tell the difference between whether it's a pH graph or a temperature graph, apart from looking at the actual axes, is you just look at the shape. So pH is symmetrical, whereas temperature is sort of lopsided and skewing to the right side. So don't have to memorize this. This is just to show you that there's a whole wide range of pHs. You don't have to memorize this though. Maybe just memorize like pepsin is in the stomach, but you don't have to really know any of the other ones. But of course, if they tell you that the pH of the small intestine is 6.2, and they ask you for the optimal pH of an enzyme that's in the small intestine, then you would say, well, around about 6.2. Cell signaling, I'm not sure if anyone else is going to be covering this in later lectures, but I'll just briefly talk about it. So cell signaling, contact dependent is when two cells directly touch, for instance, your cytotoxic T cells and their target cells or other immune cells interacting. Synaptic is only between neurons, so that's using neurotransmitters. Paracrine is when your signal isn't directed at like a specific cell, it's just sent like into neighboring cells, so all the neighboring cells will receive it. Slightly different from synaptic because it doesn't occur between a synapse, because paracrine can occur between non-neuron cells, but technically a neuron signaling is also paracrine. So synaptic is just a more specific term for it. Endocrine means it has to go through the bloodstream. That's why the endocrine system, the hormone system, is called endocrine, because hormones are produced in glands and then they go through the bloodstream to their target organ. Autocrine means that the cell is going to signal itself. So it's very blurry, but just a diagram for you guys to look at a bit later. So moving on to some macromolecules. So this is a little bit of revision, some new stuff. Carbohydrates. So know the function of carbohydrates, know the names of them. So you probably would have seen glucose, fructose, galactose, lactose, maltose, sucrose, starch, glycogen, cellulose in year 12 biology. This might be a new one. So an oligosaccharide is 3 to 10 joined monomers. So it's bigger than a disaccharide, which is strictly 2 monomers, and slightly smaller than a polysaccharide, which is like lots and lots, up to thousands of monomers. So this is sort of like the little intermediate. So if you see this, just make sure you know that it's the intermediate between a disaccharide and a polysaccharide. You should know the functions of them. So for semester 1, what they'll usually be testing you on is how they are used for energy and for energy storage. So glycogen, which is a polysaccharide, is storage in your muscles and in your liver as a source of glucose. But you should also know that you have different carbohydrates that come from different sources that can also do different things. So the polysaccharide cellulose is actually dietary fiber, which will provide bulk for the diet, and it will also help to clean your gut as it moves through. It will aid the synthesis of other substances. So if you've got glycolipids and glycoproteins, the glyco indicates that you've actually got carbohydrates that are part of that. So you can, if you don't have carbohydrates, if you eliminate all the carbohydrates, you won't actually have these being produced. Energy sparing. So carbohydrates are sort of like the first thing to be used when your body needs energy. So it will save your proteins, and we always want to avoid protein breakdown. So this is just another diagram of different carbohydrates. So these are your monosaccharides, polysaccharides. They haven't shown you maltose, but maltose is just double of that. And then your polysaccharides. Yes. Just note that this doesn't show bonding, which is on the next page. So for the bonding stuff between polysaccharides, this should be revision from year 12 again. So cellulose is in plants, beta glucose bonds. I'm not sure if, I don't remember if you guys did that, but just memorize that cellulose has beta glucose bonds. Therefore, we can't break down cellulose because we don't actually have the enzyme to break down this bond. And it's very tightly bonded. So that's why we can't, another reason why we can't break it down. Starch can be broken down into amylose and amylopectin. So amylose is like slightly more linear than amylopectin, which is branched, but glycogen is the most branched. So the order is cellulose, amylose, amylopectin, glycogen for order of branching, for number of branching. For all the molecules that we can break down, the subunit is alpha glucose. Okay, so we can break that down. Oops, there we go. So glycosidic bonding. So this might be a new concept for all of you who did chemistry, which is, I think, everybody. So alpha glycosidic bonding means that the bonds look like this. Beta glycosidic bonding means that the bonds look like this. So essentially just recognize what the bonds look like. So alpha means they're going in the same direction. Beta means that they're doing this, is probably what you need to know. And also just knowing that between carbohydrates, it's going to be alpha 1,4, and then between different chains. So if you're bonding two different chains of the carbohydrate, then it's actually alpha 1,6, which is just telling you where the bond is happening between. So fortunately, this is just something that you just have to memorize. And I suppose the good thing is you memorize it for the exam. It is still quite low yield. So if you're stressed with other things, you don't have to worry about it, but you memorize it for the exam, but you don't actually see it afterwards. Not sure if that's encouraging or not. Second macromolecule type, lipids. So lipids are stored as triglycerides or triacylglycerols. They are essentially the same thing. They're a glycerol with 3 fatty acid chains, and they are important for many functions, which Akhil will go into later. There are also lipids that are not used for energy or are not meant to be used for energy. So phospholipids, they have the role that of the plasma membrane maintaining fluidity. They will also help with absorbing fats and also essential for making lipoproteins, which is something that Akhil will also go into. So phospholipids have 2 fatty acid tails. Triglycerides have 3 fatty acid tails. So if you're ever confused about them, just look at how many tails they have. Cholesterol or steroids, they are sort of the same thing. So cholesterol is a steroid and can be made and can be used to make steroids. It's 4 rings. Okay, so if they tell you it's a lipid and they give you this structure, there's 4 hydrocarbon rings that are fused together with some other stuff. So most of these steroid hormones and vitamin D and stuff will also have a similar structure. Cholesterol is very important. So when you think of cholesterol, like usually you think of it as a bad thing because high cholesterol will give you bad health outcomes, but it's actually essential for body function as well. Apart from maintaining the membrane fluidity, it's actually a precursor to many other things. So steroid hormones such as testosterone, cortisol, bile acids, so that's used for digestion. Vitamin D is sort of synthesized from cholesterol in your skin, and what I said before about membrane fluidity. Okay, so just knowing that you can also have a mixture of these carbohydrates and proteins, or carbohydrates and lipids, to make glycoproteins and glycolipids. So with this, just knowing that they exist, knowing that they're used usually for cell signaling, and that glycolipids can be used in blood type recognition. So the glyco part is sticking out of the cell membrane and is usually used for signaling, whereas the protein and the lipid is part of the cell membrane that keeps it sort of stuck onto the cell membrane. So if you want a carbohydrate in the cell membrane, you stick it onto a protein or a lipid. Okay, so a bit more information about phospholipids, a bit more information about cholesterol. Okay, so know some functions of proteins. So this is essentially assumed knowledge from year 12 or whenever you guys did biology. It might have been the bridging course, but just know a couple of important proteins, like know hemoglobin, because that can be tested in multiple different ways. and know like how to recognize an enzyme. Apart from that, you don't really need to know anything in particular, apart from the names of all of the enzymes involved in metabolism. So that will be in the next lecture. Proteins shouldn't be used for energy unless in starvation. So when you're starting to use your proteins and doing gluconeogenesis, then that's quite dangerous for the body. Protein structure. So this is actually slightly more high yield than the rest of the stuff as well. So remember, everything in this lecture is like, apart from the acid-base stuff and the body fluids bit, is it's not really going to be tested directly. It's just assumed knowledge. So protein structure, primary means your sequence of amino acids. Secondary means you're folding it locally. Tertiary means that you're folding it into a glob, like it's a 3D shape, and some proteins will end at tertiary structure. Quaternary means that it's a more complex protein that requires multiple chains of amino acids in order to make a single protein. So if they give you a picture, it won't be like this. If they give you like a picture that you can see there's beta pleated sheets and alpha helices, it's quite uniform, then you would say it's depicting secondary structure if it's zoomed in. If they give you one of these and you can see the alpha helices and beta pleated sheets, then you would say it's tertiary structure. If they want to give you a quaternary structure protein, then they might put it in different colors, or they'll make it quite obvious that there's two different chains, or they'll give you some hint in the background of the stem of the question. So a little bit of metabolism stuff. So energy values. This is just for you to memorize. You probably don't have to know the actual value, but you do have to know the order. So fats will obviously give you the most energy, most kilojoules per gram. Alcohols, carbohydrates, and then proteins are quite similar. So yeah, so that's good because that means your fat is a buffer system for proteins breakdown in the body, but it's also not good if you're trying to lose weight. Okay, just some terminology that I was struggling with, mainly this bit, when I was doing semester 1 last year. So mainly like glycogenesis and gluconeogenesis, they're both making glucose, but the source is different. So oh, I've spelled that wrong. Okay, so glycogenesis is synthesis of glycogen from glucose. So what I was confused about was glycogenolysis and gluconeogenesis. So glycogen lysis means you're lysing the glycogen, which means you're breaking it down into glucose. Gluconeogenesis is you're generating glucose from something that previously wasn't a carbohydrate. So things like fatty acids and amino acids, if you're generating glucose from those, it's called gluconeogenesis. If you're breaking down glycogen, which is a carbohydrate, into glucose, that's glycogenolysis. Yes. Okay. Digestion and mobilization, just something to fill the space. Digestion is when you're breaking down stuff in the diet, and mobilization is when you're taking stuff that you've stored up, things like fats and carbohydrate, glycogen storages in your muscles and your liver. That's mobilization. So digestion is new stuff. Mobilization is stored up energy sources in your body. So now time for a quiz. I'm not sure how much time we have left, so I might just go for 3 minutes. Okay, so which statement about the structure of polysaccharides is correct? So just have a read of this. Have an answer in your mind. Okay, so the answer is 3. So in amylopectin molecules, monomers are connected by alpha 1,4 glycosidic and alpha 1,6 glycosidic bonding, and that's because amylopectin has links between chains. So remember, links between chains is alpha 1,6. Links between the actual monomers in the same chain is alpha 1,4. And also just noting that it's alpha, not beta, because alpha bonds occur in between molecules that we can break down. Beta bonds are actually between cellulose. So that's the one that we can't break down. So because amylopectin is a type of starch, we have to be able to break it down. So it's alpha. Number 2, I'm not sure if anyone actually picked this one. That was like the next the next one that I thought people would pick. So glycogen is not structurally similar to amylopectin. That's incorrect because they both have quite intense bonding. Glycogen is just slightly more, so they are still considered structurally similar. If they said glycogen is not structurally similar to amylose, then that might be a better one for you to pick. But they are structurally similar because they both have these alpha-1,4 glycosidic bonds between the monomers on the same chain, and then alpha-1,6 glycosidic bonds here. And they're also both made up of glucose monomers, so they are very similar. This one, lactose, maltose, sucrose, hopefully you guys all know that they are disaccharides, not monosaccharides. Next one. So which level of protein structures hemoglobin? It's quaternary. So this is just something that you need to memorize. It's quaternary structure because it's got 4 separate chains. And there's our iron. So iron is an inorganic cofactor. Patient enters the ED severely dehydrated. Which solution could be given intravenously? So we're not going to pick urea because urea is going to kill that patient, or not be good for the patient, might not kill them, but we don't give it in like clinical situations. 9% saline, similar to 0.9, but that's actually not 0.9% saline. So that's actually not going to be good for the patient either because you're injecting a hypertonic solution, which is going to cause your cells to shrivel up because water is going to be drawn out of the cells, and it's actually going to not really treat the dehydration. It's going to make your cells dehydrated, even if your extracellular fluid is replenished slightly. 5% saline. So again, this is just trying to trick you because if you don't know the percentage of saline to give, you might get it mixed up with the percentage of dextrose that you're meant to give. So the answer is A, because everything else, apart from E, everything here, B to D, they have the wrong percentages. E is just not clinical, so it's going to be A. Okay, and dextrose is just glucose, which will eventually add to both the extracellular and the intracellular fluid. Red blood cell is placed in a solution and it is lysed. What could that solution be? So for this one, we want to think about what the effects of these are. So A, 0.9% saline, that's not going to lyse your red blood cell because this is isoosmolar and isotonic, so it's not actually going to do much. It's just going to stay in the extracellular fluid. 9% saline, that is a lot larger than 0.9% saline. That's actually going to be hypertonic. Hypertonic, which means the cell shrivels. 1% dextrose is hypotonic, which means there's less dextrose or less non-diffusible solids on the outside of the cell, which causes water to go into the cell to try and balance that out. Okay. I think I've gone over time, so I'm going to stop this. All of the solutions and stuff for the rest of them are going to be on the posted. I think they're already posted. So last thing is, if you have any questions or if there's anything that you want me to clarify, or if there's anything that you think that I said incorrectly, feel free to email me or Facebook message me. And if there's anything wrong, I'll get Akhil to maybe post it on your Facebook page. So if you ever see me in uni, come and say hi because I don't think I'm very scary. Okay, see you. Good luck for the exam. I think it's next semester, and good luck for the rest of the year. So congratulations, first of all, on getting into med, and second of all, of finishing probably the most difficult semester of year 1 medicine that we've ever experienced. So you should be all very proud of yourselves and just take it easy with revision.
Thank you so much, Callie. That was a really good presentation. All right, so I'll just be sharing my screen now. We'll get started on metabolism, which is what I'll be covering, and then after that we'll have a break. So whenever I end up finishing, so probably about 12:05, 12:10. Metabolism is a pretty long topic, and then we'll have like a 20-25 minute break till about 12:30, and then we'll go on with, I think, HKS and the rest of theme 2. All right, cool. Let me just share my screen. Here we go. There we are. Okay, if there are any questions at any time, feel free to just chuck it in the chat. I'll just keep the chat open. There we go. Oh dear, done. There we go, and I'll just present. Oh, okay, cool. Can everybody see my screen? Everything okay? I'm gonna assume that's yes. All right, so let's get started with metabolism. So just starting off with some acknowledgements of where I got some of the content from. So a lot of it was from last year's revision slides, NERMA slides, and metabolism revision doc that I made. If you want to see, I made a massive metabolism revision doc last year for everyone in year one, which can be found in the year one 2019 folder under, I think, revision documents. I made one for metabolism, neurophys, and then some other ones for semester 2. So feel free to check those out along with obviously the stuff on my OneNote. I think, pass them around to you. So yeah. Okay, cool. So this is just going to be a general overview of what I'm going to be covering. So firstly, some definitions, then we'll go into some carbs, then lipids, a little bit on protein. We didn't end up getting taught too much about protein. I'm not sure how much you guys have been taught about it. So when we get to that, if you guys let me know how much you've covered, and I'll try and cover what I learned and try and adapt to what you guys have learned as well. Then I'll go into alcohol and then finish off with a great metabolic race. I'm not sure we'll have time for questions at the end or in terms of like practice questions, but just letting you know, metabolism is pretty high yield and I'd recommend getting some practice in. All right, cool. Let's start with some definitions. So firstly, metabolism is defined as the chemical process involving the breakdown of energy-rich substrates to yield energy, which is used for several life-sustaining purposes. Or in simple English, it's the way the body breaks down fats, carbs, etc., to make energy. So anabolism is building stuff up, and catabolism is breaking stuff down. And catabolism usually ends up producing net ATP/energy. An important note is that digestion does not equal metabolism, because digestion is simply the mechanical breakdown of food into smaller molecules, and when those smaller molecules are broken down to release energy, then that's metabolism. And then just general thing, anything ending with lysis is breaking down. Anything ending with genesis is the creation of. So when it's glycolysis, it is the breaking down of glycogen. And if it's glycogenesis, it is the creation of glycogen. For example, anything ending with ACE enzyme, some stuff there. Okay, cool. So firstly, ATP is effectively the powerhouse of the cell, which is basically broken down into ADP and a phosphate group and energy. So as you can see in that diagram, you have what's ADP bind to a phosphate group, which is ATP, and then it gets broken down to release phosphate and releases energy as a result. So food is eventually broken down, eventually converting converting ADP to ATP to form usable energy. So it's effectively the creation of energy, because conservation of energy and all, but this is mostly done via electron carriers, which I'll get into a little bit more later. So the conversion of NAD plus to NADH and then FAD to FADH2. Okay, cool. Let's start with carbohydrates. So firstly, the structure of carbohydrates. So I think Kelly spoke a lot about the different structures of the carbohydrates, so I won't cover it too long, but essentially on the left, you have glycogen, and then on the right, you have glucose. So glycogen is a polymer of glucose, and it's used mostly for storage and is formed in cells. Two of the bonds that they kind of say you need to know, or they like mention a bit, are glycosidic bonds. As implied in the name, it's bonds within the glycogen molecule. They're not exceptionally important except when they might ask a question about it using like a buzzword or something. So if you've got alpha 1 4 glycosidic bonds form branches, and the alpha 1 6 glycosidic bonds, if you guys can see a mouse forms bonds over here instead. And then there's branching about every 10 glucose. And glucose is simply a monomer, which is 6 carbon sugar in its form. And it is the form that's absorbed in the small intestine within the GIT, and it can be transported in the blood, which becomes important in terms of diabetes and maintaining blood sugar. So digestion is the breakdown of dietary nutrients into smaller components of the GIT, so they can be absorbed by hydrolysis. So mostly in our diet is disaccharides and polysaccharides, which I think Kelly covered. If you guys have any questions about it, really, you can feel free to ask me, but they need to be broken down into monosaccharides, glucose, really, to be absorbed. So disaccharides is implied by the prefix is made up of really 2 sugars. So sucrose breaks down into fructose and glucose. Lactose gets broken down into galactose and glucose by lactase. And then you also have maltose, which is broken down by maltase. And some polysaccharides is starch, glycogen, and cellulose. Starch is basically the main component of what we eat. And then cellulose is important because it can't be digested, and it makes up dietary fiber, and it consists of beta-1,4 glycosidic bonds instead, which can't be digested by the GIT. So basically, the breakdown starts in the mouth with salivary amylase, which is an important enzyme to remember, and it continues in the small intestine with pancreatic amylase. So the different amylase enzymes break down the alpha-1,4 glycosidic bonds, and in the small intestines, the epithelial cells use alpha-glucosidase enzyme, break down alpha-1,6. So this becomes relevant, the use of the alpha-glucosidase enzyme, mostly in second year in regards to diabetes and diabetes management, but for now, there's no real need to worry too much about that. So starch gets broken down to limit dextrins and maltose, and each of these are broken down by their relative enzymes. So dextrinase breaks down the limit dextrins, and then maltase breaks down the maltose. And they break it down to a single glucose unit and 2 glucose subunits, respectively. Just remember some of the enzymes, because I do remember them asking a question about limit dextrins last year. Literally just asking what breaks it down. Yeah. Then glucose subunits are effectively absorbed into epithelial cells by the microvilli on the apical membrane, which is the top part, or the part which is facing the lumen, and then it gets absorbed by a secondary active transport. And the main active transport is the sodium potassium pump, which I'm sure you guys have been drilled into plenty. And then carbohydrates are stored in the liver as liver glycogen, muscle glycogen, and then as well can be stored as adipose. So regarding the storage and mobilization, so whenever blood glucose levels are high after a meal, because of course the blood glucose is absorbed, insulin is produced to signal to muscle and liver cells to update the blood glucose and store it as glycogen, which is the process called glycogenesis. And then whereas when blood sugar is low, glucagon is released to signal to muscles and cells to break down glycogen in glycolysis. Glycogenolysis. So effectively, glycogenolysis is breakdown of glycogen to glucose, and insulin and glucagon are the main hormonal regulators of breakdown and formation of glycogen, and that's released by the pancreas. So glycogen is an important note is that glycogen is highly branched, and it gets broken down into glucose 6-phosphate and needs debranching enzyme. An important note is that in the liver, it gets glucose 6-phosphate can get broken down by glucose 6-phosphatase, which dephosphorylates it into glucose to maintain blood glucose levels. But this can't happen in any other part of the body, as there are no glucose 6-phosphatase enzymes in skeletal muscle cells. So the glucose 6-phosphate instead directly enters a glycolytic pathway for production of energy. So the glycogen which means that the glycogen stores in the muscles are effectively used by the muscles themselves instead of being broken down to glucose to maintain blood glucose levels. So in the bottom left there, I've put a little table with the enzyme glycogen synthase as implied by the name to form glycogen, and then glycogen phosphorylase to break down glycogen. and the different hormonal regulators of it. So insulin stimulates glycogen synthesis, whereas glucagon inhibits it, and epinephrine inhibits it. So epinephrine, I'm sure you guys would have covered a bit about sympathetics and parasympathetics. So epinephrine is basically the same thing as like noradrenaline. So obviously in a fight or flight, you want higher blood glucose levels because you need the energy. So you'll break down your glycogen. Okay, cool. So just another table on glycogenolysis and then glycogenesis. So glycogen synthesis is effectively done by the glycogen synthase enzyme, which adds glucose monomers along the chain. So the branching enzyme effectively also adds branches to the glycogen, which allows for more access when broken down. I spoke a little bit about insulin, but the mechanism of how insulin causes the formation of glycogen isn't as important as the fact that insulin actually does encourage glycogen synthesis. Whereas in regards to glycogen breakdown, as I mentioned, glycogen phosphorylase breaks down alpha-1 for bonds of glycogen, releasing glucose-1 phosphate, and then eventually glucose and adrenaline can stimulate this, and glucagon binding in the liver effectively causes phosphorylation of both enzymes, and then those enzymes, once phosphorylated, actually cause glycogenolysis. It's a lot of big words and it can be like a bit confusing. So if you have any questions, feel free to ask. And also just try and get the main takeaway from glycogenolysis and glycogenesis, which is a hormonal regulation, and the fact that it's different in the liver and skeletal muscle cells. So the glucose 6 phosphatase in the liver, and the purpose of glycogen breakdown to maintain serum glucose levels. Whereas in skeletal muscle cells, it's breaking down glycogen in order for the muscle to function. Okay, cool. So speaking of the formation of glucose and maintaining serum glucose levels, gluconeogenesis is effectively the process of creating glucose from non-carbohydrates, and it only really occurs in the liver. So it's mostly used during fasting or starvation when carbohydrate stores have been depleted. So the sources of the appropriate sources effectively are altered in order to form glucose, and they become a reactant in either a stage of glycolysis or the citric acid cycle / Krebs cycle. So glycerol feeds into step 5 of glycolysis when broken down. I'll cover that a little bit more when speaking about fats a bit later. Lactate feeds into pyruvate, and acetone also feeds into pyruvate. Then amino acids become pyruvate and undergo deamination and stuff like that in order to become usable energy through the glucose-alanine cycle, which I'll cover a bit later. And then acetyl-CoA, you guys should hopefully know that that's already in the cycle. But glycolysis is an important note is that glycolysis is reversible, except you can't get pyruvate from acetyl-CoA because it's kind of like a one-way reaction. So acetyl-CoA has to kind of go all the way back around the cycle and become oxaloacetate, and then work its way back up from oxaloacetate to become glucose again. So again, as I have the TLDR on the right, reverse glycolysis, and it bypasses a few steps and it occurs in the liver. So 6 ATP is used per glucose molecule synthesized, and is energy mainly provided by fatty acids. And another important note is that this is the main source of blood glucose after 8 hours of not eating. That'll also get covered a little bit more in the great metabolic race when I covered. So you know what they say about real estate, location, location, location. So these are the different processes that happen in regards to the breakdown of carbohydrates and their location within different cells. So in eukaryotes, glycolysis happens in the cytoplasm. Fermentation happens in the cytoplasm, similarly with prokaryotes. But regarding pyruvate breakdown and decarboxylation, in eukaryotes, it happens in the mitochondria, along with the Krebs cycle whereas in prokaryotes, because they don't have mitochondria, it happens in the plasma membrane and then the cytoplasm. So just know the differences between where the two are. So be sure to look at that table again later. And an important note for the ETC, electron transport chain, is that within eukaryotes, it happens within the inner membrane of the mitochondrial matrix. Okay, so let's move on to glycolysis. So as you can see on the right there, it is a very complex process, and you're not expected to learn pretty much anything really from that of those enzymes. So please don't spend too much time worrying too much about the individual names or whatever, because I know a lot of people ended up wasting a bunch of time doing that. So basically, why do we need glycolysis? Glucose contains a bunch of energy, and you need to break it down slowly and have a sustained release of energy. And this is, and most of the ATP that's produced is via NAD, is via the conversion of, and via using electron transporters. So, pardon me, electron carriers. And those get converted, those help in the formation of ATP in the electron transport chain. So key points for glycolysis, it's a 10-stage process happening in the cytoplasm of cells. So there's a preparatory phase and a pair phase. So they're two real phases. As you can see, in the being there's the conversion of glucose to glucose 6-phosphatase, which requires an ATP. And then similarly of fructose 6-phosphatase to fructose 1,6-diphosphatase, that also requires an ATP. So you need to firstly have energy to break it down, and then later on in the cycle, you get the release of 2 ATP twice. So in the last stage, and then I believe that's stage 6, if I remember correctly. But more importantly, you have the input of a single glucose and 2 ATP, and you end up with 2 pyruvate, 4 ATP, 2 NADH. Sadly, the numbers you guys actually kind of need to know, or like that's something you have to memorize, basically, literally for the purpose of the exam. Otherwise, there's no real use for it, I'll be honest. Okay, cool. The fate of pyruvate. So, this is depending on the presence of oxygen. So this is basically the pyruvate decarboxylation that I mentioned earlier. So regarding aerobic respiration in total, this is not from the formation of just pyruvate, but this is like in total, aerobic respiration produces a total of I have 36 to 38 ATP per glucose molecule. It can range from 32 to 38 depending on the textbook that you read. So essentially, pyruvate is converted to acetyl-CoA with the release of NADH, and it'll later enter the citric acid cycle. Some important notes about it is that it's catalyzed by magnesium. It's a spontaneous reaction, so if you guys remember your chemistry, that's a negative delta G. It uses the enzyme pyruvate dehydrogenase, important enzyme to remember. And the enzyme requires thiamine, which is a group B vitamin. That's also important. That becomes a lot more relevant when speaking about alcohol metabolism. So people who are alcoholics often have have the thiamine deficiencies. You'll learn more about that in second year in regards to GIT and absorption of the B vitamins. But long story short is they lack thiamine, and then they lack the ability to undergo aerobic respiration, leading to more anaerobic respiration along with other reasons to do with NAD plus and NADH. So for the input of aerobic respiration, don't worry, I've got a chart of it, I think, on the next slide. So you have 2 pyruvate and 2 NAD plus, and this is per molecule of glucose, by the way, and ends up with an output of 2 acetyl-CoA, 2 NADH, and 2 CO2. Now, speaking about anaerobic respiration, which is also known as fermentation, so that occurs in the cytoplasm of cells, pardon me. And it's effectively when pyruvate is metabolized to lactate or lactic acid to release NAD plus and eventually to net ATP. But so the lactic acid and lactate is eventually transported to liver, and it gets regenerated by the Cori cycle into glucose when the anaerobic conditions are met. So I've got a slide on that. The Cori cycle isn't too important. All you need to really know is that lactic acid eventually, when the body is capable of undergoing aerobic respiration again and is no longer producing a stupid amount of lactate, it can get recycled into glucose. And the breakdown of pyruvate into lactate uses the enzyme lactate dehydrogenase, important enzyme to know, and this is at a high level if glucose isn't stored properly. And the buildup of lactate is also very dangerous and very clinically relevant because it can cause a lactic acidosis, which it means that your blood pH is too low, and that can lead to a lot of problems. So the inputs for that are 2-pyruvate and 2-NADH, and the NADH gets converted into NAD+, and this ends up with the output of 2-lactate and 2-NAD+, and this is per molecule of glucose. And then in yeasts, for some reason they bring up yeasts, it gets converted to ethanol and CO2 instead of lactate. So, yeah, cool. I've got questions that I meant to say when aerobic conditions are met regarding Cori cycle. Yeah, probably. Thanks for pointing that out. I'll adjust that in slides. I'm sorry. Okay, so these are some really good diagrams that Sophie Hiscock, she's year 3, she drew them. They're really good. I'm useless at art, so I just snagged them off her. So on top left, you have anaerobic respiration, the conversion of pyruvate to lactate. As you see there, converting NADH plus to H plus. To NAD+, in this process, 2 hydrogen atoms are added to the central carbon. On the right, you have aerobic respiration, which is when you effectively start with pyruvate, and then with the help of coenzyme A, you end up with a release of CO2, the conversion of NAD+ to NADH, and you essentially end up with acetyl-CoA and CO2, isn't it? And then the bottom there is a little thing on the Cori cycle, which is recycling lactate to glucose. For those of you who are more interested in it, it's not exceptionally important. But as you see here on the right of the cycle, you have the conversion of glucose to pyruvate to lactate, which is anaerobic respiration. And eventually that gets the lactate stays in the blood. And when possible, the lactate gets converted back to pyruvate and then back to glucose, requiring ATP and essentially undergoing gluconeogenesis. This happens within the liver, of course, because gluconeogenesis only really happens in the liver. So, yeah, there we go. Okay, now a much more fun part. Let me just check the time. Okay, the Krebs cycle. It's a major contributor to depression in year 1 med students, but more importantly, it's the major bulk of ATP, NADH, FADH2 release. So it essentially creates a lot of energy through creating a lot of different electron carriers. So what you need to know about it, I've tried to keep it blunt, is that it occurs in the mitochondrial matrix of cells. It's a 10-step cyclic process. It starts with acetyl-CoA, as you can see at the top there, which enters the citric acid cycle, and eventually just keeps going around and around and around. So a lot of the individual names, you don't need to know too much about the individual names, but some of them, okay. More importantly, you need NAD plus to keep the citric acid cycle working. So with low levels of NAD plus, which can happen when you consume alcohol, because it converts NAD plus to NADH, the citric acid won't work. So even if you have a bunch of acetyl-CoA, you simply can't undergo the citric acid cycle without a sufficient amount of NAD plus. And as we see here in inputs per 1 glucose, so if you want to do it per acetyl-CoA, divide everything there by 2. But so you end up with 2 acetyl-CoA. Your inputs are 2 acetyl-CoA, 6 NAD plus, 2 FAD, 2 ADP, and phosphate, and then the outputs end up with 4 CO2, which is ends up being 2 CO2 per Cl-CoA, 6 NADH, which is produced in steps 3, 4, and 8. So as you can see here, that's 3 per acetyl-CoA, 2 FADH2, or 1 per cycle, or 1 per acetyl-CoA, which is produced in step 6, and then you end up with 2 GTP. This is per glucose, not per acetyl-CoA, so just remember that. It's important to just remember some of the steps where the electron carriers are produced, and it's important to note is that GTP is produced instead of ATP. They have effectively the same use, or like they're effectively the same thing for the basis of the exam, but you just need to know that they're different, but they do the same purpose. Okay, so the electron transport chain, as you see here, it is a very funny, scary diagram, but I've tried my best to break it down. So I'll swap between the first and the second slide, or this slide and the next slide. So, okay, this is work. Okay, great. So the electron transport chain, also known as oxidative phosphorylation, is where the electron carriers, so FADH2, NADH, are transported, and they will gradually release their energy. So the way that they actually create their energy is through the formation of ATP and the creation of a proton motive force, which is used by the ATP synthase enzyme to form ATP. So, of course, the electron transport chain can't happen without oxygen. So, this takes place in the inner mitochondrial membrane, the cristae. And so, essentially, the mitochondria has two membranes, the inner and outer membrane, and the space in between the two is intermembrane space. So let me just go back here so you can see the outer mitochondrial membrane there. You can see the intermembrane space, which is where the hydrogen gets pumped into. And here you have the inner mitochondrial membrane, which is where all the different like enzymes and stuff are, and the ATP synthase stuff. So I'll come back to this diagram again in a second, but I'll firstly go on to the different complexes that are important in the electron transport chain. So first we have NADHQ reductase. So this is, you don't need to know the name, just know that this is the first complex really. And this is where NADH plus arrives to and effectively moves hydrogen plus ions into the intermembrane space. So as you can see here, NADH comes in, it releases its hydrogen and gets converted to NAD plus, and the hydrogen gets pumped out into the intermembrane space. Then next you have succinate dehydrogenase, or more importantly known as the second complex, which is where FADH arrives to. And with the second complex, there's a little thing called ubiquinone, which is basically an electron transporter within the complex, and it transports electrons from the succinate dehydrogenase and NADHQ reductase to complex 3, which I'll get to in a second. Hold on. So let me just go back a second. So as you see here, you have NADHQ reductase. The hydrogen's gone pumped into the intermembrane space. The second one, what's at the side of the succinate dehydrogenase. The second complex is where the FADH2 arrives, and eventually the electrons, as you can see here, get transported through the ubiquinone and eventually go on to the third complex, the fourth, and then the fifth, and then back out later, which I'll speak about later. Then you have your cytochrome reductase, which is another complex, and then cytochrome C is also in there between the complexes 3 and 4, and that's also an electron transporter. Then you have cytochrome oxidase, which is where all the electrons that they've generated, or all the different electrons that have been processed, are used to convert oxygen to water. So that's where all the electrons actually go. And during this whole process, keep in mind that hydrogen is constantly getting pumped into the intermembrane space, which is creating a difference in the electrostatic charge. So of course, it'll be a buildup of hydrogen over there in the intermembrane space, and a positive charge over there that becomes important in regards to ATP synthase and the proton motive force. So, important note on cytochrome oxidase is that cyanide inhibits this complex and thus can inhibit the electron transport chain and cause anaerobic glycolysis instead. So that's just useful for whenever they ask a question about what does cyanide inhibit or where does cyanide act, because that's a common question to add like. So just remember cytochrome oxidase. Now lastly, the last part of the electron transport chain is the ATP synthase. So this is the important enzyme. So basically the potential generated by moving the hydrogen ions into the intermembrane space is used, and that proton motive force is used by the ATP synthase to effectively move the hydrogen down the concentration gradient. And by moving all the hydrogen from the intermembrane space back into the cell, this effectively converts ADP into ATP, and that's important. And that essentially is how you correct the formation of ATP through the electron transport chain. An important note is that each NADH molecule pumps 10 hydrogen ions into the intermembrane space. And because it is 4 hydrogen ions per ATP, or 4 hydrogen ions required to phosphorylate ADP into ATP, each NADH molecule ends up producing 2.5 ATP, and then each FADH2 molecule, which pumps 6 into the intermembrane space, ends up producing 1.5 ATP. And another important note is that whenever tissue is deprived of oxygen or is hypoxic, the electron transport chain is inhibited, and the amount of ATP generated decreases substantially and leads to anaerobic respiration to produce lactate. And an important note is that the cells which are hypoxic, they start swelling up. Okay, cool. So here's just basically the maths of how you end up creating, or like how much ATP ends up creating. So for glycolysis, you end up per glucose creating, or with the formation of 2 NADH and 2 ATP, so 2 times 2.5 ends up with 5 ATP from the NADH in glycolysis and 2 ATP there. Regarding pyruvate decarboxylation, per glucose and forming 2 NADH, and that ends up being 5 ATP there. Then citric acid cycle, as you can see, where the bulk of the electron carriers are made or formed. There's 6 NADH, 2 FADH2, and 2 ATP. So 6 by 2.5, 15, 2 by 1.5, 3, and then extra 2, you add them all up together and you get 32 ATP. There we go. Okay, now that's what we have with carbohydrates. So that's one of the big parts down. Now we're moving on to lipids and fats. So, Callie covered this in a lot more depth, I assume, but just to lightly touch on the different lipid structures. So there are 3 types of lipids, all of which are nonpolar, and the fact that they're nonpolar is important because they can cross cell membranes. So you have your tags, which are also known as your triacylglycerols or your triglycerides. This was something that was pretty confusing for me, which was that glycerol and tags are different, and that every single thing that's triacylglycerol, triglyceride, they're alluding to the same thing. And so tags are made up of 3 fatty acids, as you can see, the 3 fatty acids, all bound to a glycerol backbone, therefore are forming the triglyceride. And that becomes important when in terms of breaking it down. You also have phospholipids, which is 2 fatty acids, and those are mostly used for cell membranes and neurotransmitters. And then you have sterols, which also mostly are steroids and like cholesterol, and they're precursor for bile acids, vitamin D, and other hormones. And they're also kind of used for membranes. Yeah, and you can see the structure on the bottom right there. So moving on to the types of fatty acids. So you have saturated, monounsaturated, and polysaturated. I think that's what I say, polyunsaturated, sorry. Fatty acids. So you have you can either have fatty acids which are saturated or unsaturated, and unsaturated are the ones with the double bonds. It's just how to remember. And you remember, hopefully you still remember that a little bit from chemistry. But moving on to the essential fatty acids, which are have to be obtained from your diet, as they can't be synthesized by your body. So these are really important in cell membranes and for eicosanoids. Norma probably would have covered this as well, but this becomes pretty clinically relevant later and is an important part of like, you know, the whole omega-3, omega-6 craze that people have with their diets is because of this. It's because it's really important in inflammation and the formation of arachidonic acid, and actually the precursor and formation of eicosanoids. So eicosanoids are essentially paracrine hormones. So if you remember paracrine, that's type of cell signaling, and they're essentially prostaglandins. If you guys have ever heard of prostaglandins, they're essentially an inflammatory mediator, and they're really important. So the prostaglandins are important in inflammation, blood clot formation, smooth muscle relaxation and contraction. So they're actually really important molecules. You'll cover them a lot more, actually, I think, in second semester when you cover pain and the different pain mechanisms. So regarding the two types of essential fatty acids, you have your omega-3s and then you have your omega-6s. So your omega-3s are basically your linolenic omega-3s, and they're found in the membranes for bilayer structures. And then you also have your omega-6 families, which are made up of linoleic omega-6 and linoleic acid. So arachidonic acid, which is a precursor for prostaglandins, they're synthesized from the linoleic acid. So in summary, you need to have a sufficient amount of these in your diet in order to have enough linoleic acid and linolenic omega-3 in order to form the arachidonic acid, which can then be used to form eicosanoids and then prostaglandins. Okay, hope that's not too confusing. Now, moving on to the digestion and absorption of fat. So dietary fats can't actually be absorbed kind of like how glucose can be. So they need to be emulsified first, and they get emulsified into tiny droplets called micelles through the process of bile, which is secreted by the gallbladder and the pancreas. So you have sorry, not the pancreas, bile by the gallbladder. So this happens in the GIT. So pancreatic lipase hydrolyzes the fat in micelles to produce fatty acids. So after the micelles, as you can see in the diagram on the right, once the big fatty acid droplet has been emulsified and put into little small micelles, they can then be broken down by pancreatic lipase and then absorbed as they'll become monoglycerides and free fatty acids. So the fatty acids and monoglycerides enters the cells via diffusion, and they enter into the enterocytes. So the fatty acids and monoglycerides are resynthesized into triglycerides, tags, inside the cell and in the smooth ER of that cell and moved into the Golgi apparatus for packaging. These tags are essentially packaged into lipoproteins called chylomicrons. This is important. Chylomicrons are actually like super important for some reason. Yeah. Well, they are really important, but you'll need to know them for your exam quite a lot. But they essentially get packaged in chylomicrons, and the chylomicrons enter circulation via they exit the enterocytes via exocytosis, and they enter the lacteals of the lymphatic system, and then the lipids via the lymphatic system are transported in the blood in chylomicron. So essentially you have chylomicron and you have tag inside of it, and that's enters circulation by the lacteals. And this is of course happening inside, like the absorption of all this is happening through microvilli and villi. So you have the breakdown in the GIT, then most of the absorption happens in like the small intestine and large intestine. Yeah. Okay, so just speaking a little bit on storage. So adipose can take up FFAs from blood, but not glycerol, and synthesize glycerol from glucose to create tags. Because of course a tag is 3 fatty acids, 3 FFAs, and a glycerol backbone. And then the liver can take up FFAs and free glycerol to recompose into triacylglycerides for storage, creating fatty livers. So the mobilization of tags and glycerol is important, and they're different. So you've got a diagram on the next slide, I believe, about it. Yeah, okay, I'll also go through. I'll just read through it first, and then I'll point it out on the diagram after. So regarding the mobilization of stored triacylglycerides within adipocytes, so this is how the tags within adipocytes and fat cells are essentially released. So glucagon is essentially released as a response to low blood sugar, and this causes or initiates a reaction or cascade of reactions to break down triacylglycerides within adipocytes. So glucagon binds to its receptor on the adipocytes and essentially creates it activates a G protein, and this through adenylate cyclase produces a secondary messenger called cyclic AMP. The whole G protein adenylate cyclase enzyme process you don't need to know. Just remember, glucagon binds and produces CAMP, and eventually CAMP, which is this secondary messenger, undergoes a lot more events and eventually activates the lipase enzyme. So the lipase enzyme within the adipocyte cleaves fatty acids away from the tags, and these free fatty acids are then released from the adipose tissue into the bloodstream with a carrier of albumin. So albumin is a protein which the liver produces. And so while bound to the albumin, the fatty acids enter the target tissues. So the target tissue can be like a muscle cell or the heart. And it enters the cytosol of that tissue via a receptor and dissociates from the albumin, therein providing free fatty acids for that target tissue to use and then break down. Now, that was the mobilization of the tags, and you know where the free fatty acids go. But now speaking about the glycerol, which gets cleaved off by a lipase, the glycerol uses glycerol kinase with ATP to get phosphorylated, converting NAD plus to NADH. So you remember how I mentioned before how glycerol can be used in gluconeogenesis. So this happens in the liver, and eventually it results in the formation of an intermediate called dihydroxyacetone phosphate. You don't actually need to know how glycerol is converted to this intermediate because it's a really long multi-stage process, but you need to know that glycerol is converted to this, and then this intermediate directly enters the glycolytic pathway in step 5 of glycolysis. And of course, in order to form, and of course, instead of getting broken down in and going into the citric acid cycle, it gets converted back up to glucose. Yeah, hopefully that makes a little bit more sense. But now with the diagram will probably explain it a little bit better. So in the left there, you can see you have the tags inside the adipocyte, you have the lipase, which breaks it down, and you have fatty acids, which are entering the bloodstream. In the adipose tissue, you have glycerol. Glycerol goes into the liver and undergoes that reaction to form the intermediate dihydroxyacetone phosphate, and that is used to form glucose, and glucose is then secreted into the blood to maintain serum glucose levels. Then the fatty acids can get broken down in the liver, or they go straight to like the heart or the target tissue where they end up getting broken down. I'll speak more about the breakdown of fatty acids in just a second, literally right now. So the breakdown of fatty acids happens through beta oxidation. So this is an important pathway. So this is essentially when you have fatty acid. So let's say they're 16 carbons long and they're broken down into acetyl-CoA, and this is through a repetitive process where they essentially cleave off 2 carbons at a time from the fatty acids. And if you remember correctly, you end up, each acetyl-CoA is 2 carbons long. So the process gets repeated 7 times to completely break down the 16 carbon long fatty acid. And an important note is that it gets repeated 7 times, we end up with 8 acetyl-CoA. Hopefully you guys understand how that works. Let me know if that's confusing, but essentially because you cleave off 7 times, you end up with 8 acetyl-CoAs, right? Okay, so you end up, so let's say your inputs are a fatty acid, 7 FAD plus, 7 NAD plus, 7 H2O, and 7 coenzymes, the 7 coenzyme A, and your outputs end up with 8 acetyl-CoA, 7 FADH2, 7 NAD plus, NADH, and 7 H plus. So as you can tell, that is a lot of energy. If you guys remember correctly, like it's 2.5 ATP per NADH, and then 1.5 ATP per FADH2. And then you have the acetyl-CoA as well. So, and that forms a bunch of electrons. So really you have the formation of tons and tons of energies from the breakdown of a single fatty acid. And then I mentioned there at the bottom there, the conversion of the glycerol backbone, but I think I spoke a bit about that before. Now, moving on to the reverse reaction of that, which is fatty acid synthesis, which is also sadly important. They like to ask you a bit about this. So this requires acetyl coenzyme A, and this mostly happens in the liver cell cytosol. It can happen in other tissues, but an important note is that adipose is not a major side of it. So this mainly happens when there's excess energy regulated by nutritional status. So if you have so if you have tons of carbs, low fat, you'll have a high rate of fatty acid synthesis, right? Because you've got excess energy and it needs to get stored. So here, there's the example of the synthesis of palmitate, which is a 16-carbon fatty acid. So if you guys remember from the last slide, I used an example of breaking down a 16-carbon fatty acid. But in this process, you have acetyl-CoA, which is converted to malonyl-CoA. This is an important step. It requires an ATP, and this is the committed step. So malonyl-CoA is converted into fatty acids, and it uses, instead of NADH and NAD+, it uses 2 NADPH. Realistically, it's not that important, but just know that it uses NADPH and not NAD+ and NADH. I won't get into why or whatever, because it's just unnecessary. And the synthesis of the fatty acids happens in the same way as a breakdown does, in the elongation of 2 carbon units at a time to form acetyl-CoA. And this uses an enzyme called fatty acid synthase to convert the malonyl-CoA into the fatty acids. And that's essentially a large multi-enzyme complex with 3 catalytic domains. You just need to know it's a big complex and it's got 3 different domains for it. So for the input to create a single palmitate molecule, you need 8 acetyl-CoA, 7 ATP, and 14 NADPH. So that's also a lot of energy. Or it starts off with a lot of energy and gets stored in a small little thing. So the general formula to produce fatty acid with an X amount of carbon atoms would be denoted by there. So X by 2 acetyl-CoA, so 16 divided 2, 8. Then you have X divided 2 minus 1 ATP. So again, I've done the maths there. And then X minus 2, so 16 minus 2, 80 plus NADPH. It's better to just memorize the formula if you really care enough. An important note is that there's a ton of regulation in regards to fatty acid synthesis to prevent excess lipid accumulation. So you don't need to know the specific steps, but you need to know that there's feedback inhibition, there's allosteric activation, there's activation of hormones, there's a rate limiting step. There are tons and tons of different aspects to try and regulate fatty acid synthesis to try and avoid excess accumulation. As you can see in the thing at the bottom right there, you don't need to know the exact process, you just need to know that that there's a lot of regulation. So regarding I'm going to try and speed up because I think I'm taking a bit too long. So this, in regards to the synthesis of triacylglycerides, you have tags which are synthesized in the liver and adipocytes, and from glycerol in the liver. So the liver synthesizes fatty acids and packages them onto the glycerol backbone to make tags, and they can get transported as VLDLs. So hold on, let me go back to here. So if you see in this diagram here, you have fatty acids converting to tags and then carried via VLDLs into the heart and into target tissues where it gets reconverted. All right, cool. And okay, there we go. And then from glucose in adipocytes, so the adipocytes absorb the glucose in the blood, and the fatty acids change from the extracellular environment and then convert them into tags for storage. And this only happens in the fed state. So when you have excess energy, tags are a great way to store energy because they're very stable. Regarding the synthesis of phospholipids is not that important, but we just need to know all animal cells except red blood cells can synthesize phospholipids, which are important for cell membranes, and this also usually happens only in high energy times. Regarding the synthesis of ketone bodies, They're released during the beta oxidation pathway too, so ketone bodies are released, and they're essentially synthesized from acetyl-CoA and fatty acids. So you have acetyl-CoA and fatty acids, and through HMG-CoA synthase, you get the production of ketones bodies. So this is important in regards to diabetes because when there's untreated diabetes, you have improper glucose uptake regulation and an increased amount of fatty acid oxidation instead to try and meet the energy needs. And this essentially results in a bunch of acetyl-CoA, which gets converted to ketone bodies because the body needs energy somehow. And ketone bodies are a sign of either diabetes or severe starvation. So let me just show you back in the diagram. Here you have fatty acids convert to ketones. Ketones can be used by muscle, brain, and the heart. I'll speak a little bit more about the tissues and what they use later. Okay, cholesterol, oh dear, cholesterol and cholesterol synthesis. So cholesterol is a hydrophobic molecule and it's essential for cell membranes and hormones and tons and tons of different things. I know it gets a bad rap, but it's really, really important. So the synthesis of cholesterol originally starts from acetyl-CoA, which gets converted into beta-hydroxy methylglycerol-CoA. That's kind of irrelevant for now, but important later in second year. But you essentially have that which is converted to mevalonate. The name is not important, but you need to know it is a committed step, and it is used. It used the enzyme HMG-CoA reductase, and inhibitors of HMG-CoA reductase, also known as statins, are really widely used in order to prevent high cholesterol or hypercholesterolemia. So that enzyme is tightly regulated by insulin and glucagon. So insulin stimulates it and glucagon inhibits it. So insulin increases the creation of cholesterol. So there usually is a balance between the loss of cholesterol via fecal excretion and absorption through de novo synthesis. I'd say, do you need to know what that means? Not really. Do you need to know those words? Probably, because you could get asked about it. And an important note is that cholesterol can't be digested or metabolized, and it's transported by lipoproteins. Now, going on to lipoproteins. So lipoproteins are essentially large molecules with a hydrophobic core and a polar coat. So essentially you have on the outer membrane apolipoprotein, you have a tag inside of it, a phospholipid membrane, and then you have cholesterol esters inside as well as cholesterol on the surface. So apolipoproteins are small protein particles on the external coat of it, and they're basically ligands for taking up lipoproteins whenever they reach a target cell. So they effectively control the metabolic fate of the lipoprotein because they are the receptor. They act as the ligand for the receptor whenever they meet a cell. So chylomicrons are a type of lipoprotein, and they're synthesized in intestinal cells after a meal, and they transport triacylglycerol and cholesterol from the intestine to the tissues after a meal. That's important. And chylomicrons are very, very high in triacylglycerol content. And once they are done with their job, they end up getting converted to chylomicron remnants and are removed by the liver. This is a pretty good slide because you need to know the different lipoproteins and the different things that they carry in their relative amounts. So chylomicrons are very good in terms of triacylglycerol transport, as I spoke about in the digestion of lipids earlier. You have your VLDLs, very low density lipoproteins, which are also synthesized by the liver, and they transport triacylglycerol from the liver to the tissues in between meals. So again, I'll show you that one diagram again, which is really important. You have triacylglycerol, your VLDLs transport them to the different tissues, as you can see here, to the heart and then to the muscle. And so they carry your tags. Then you have your low density lipoproteins. So these are also known as your bad cholesterol. So they carry cholesterol to the tissues, especially the adrenal glands or to the liver, where cholesterol is needed for the synthesis of things like steroids and hormones and stuff like that. So LDLs are the main carrier of cholesterol, and they're involved in the development of atherosclerosis and artery clogging. HDLs, also known as your good cholesterol, are protein rich and they contain pretty little cholesterol. and they transport cholesterol away from tissues. So LDLs carry cholesterol to tissues, HDL carry them away, and they bring it back to the liver and effectively prevent the arteries from high cholesterol. Okay, cool. So you see here, this is a really important diagram. I'll come back and allude to it as I explain what's going on. But normal probably would have shown this to you, and it gives me a little bit of PTSD, I'll be honest. Okay, so firstly, after consuming a high-fat meal with an enterocyte, I spoke about how fatty acids are packaged into tags and transported via chylomicrons into lymphatics. So when you have a high amount of chylomicrons, high amount of tags, the lining of capillaries have lipase, which acts upon them, on the chylomicrons and the tags, converting them into free fatty acids and glycerol. And the free fatty acids are transported to different parts of the body. So as you can see here, in the intestine, I believe it's in the intestine here, you end up with chylomicrons getting transported here, and then being released as free fatty acids into the bloodstream, and the chylomicron remnants go back to the liver. So these free fatty acids then go to the tissues. Okay, so while the chylomicrons are transported back to the liver via endogenous pathways, those remnants are converted to VLDL and then IDL and then LDL. And the LDL is transported everywhere in the body. So an important note is that when you have excess LDL as a result of excess tags and excess chylomicron remnants, you have macrophages which phagocytose the LDL, but these macrophages have a small capacity to do so. So when there's an excess of the LDL, the macrophages transform into foam cells, which just ingest LDL and stick to the blood vessel lining, which leads to a lot of free cholesterol accumulating in membranes. And these foam cells kind of stack up against each other and create the formation of cholesterol-rich plaque, which can lead to atherosclerosis, necrosis, a bunch of really, really bad stuff. So if you look back here, you have your VLDLs becoming your IDLs and your FFAs here, and your LDLs here, right? So of course you have your free fatty acids, which have been here, and the LDLs, which are transporting cholesterol to the tissue as well. But the LDLs can get uptaken by the macrophages and foam cells, and those can get stuck within the blood vessel. They'll cover a bit more on athero later as well, so no need to worry about it too much. Then all cholesterol is essentially brought back to the liver and converted to HDL, which has a low amount of protein. And HDL goes through the blood and has an enzyme which esterifies the cholesterol upon contact. And basically, the HDL uptakes the cholesterol and brings it back to the liver. Oh dear. There we go. Yeah. So you see here the LDL going back via endogenous pathway, coming back into the liver, and the HDL exiting and going to the foam cells and macrophages, collecting cholesterol, coming back. Okay, great. So LDL uptake, this is important to know, that the LDL particle with cholesterol in it approaches the liver. So this is in regards to taking back cholesterol and bringing it back into the liver and bringing it essentially into cells. So the LDL particle, of course, has an apolipoprotein on the outside, and as this receptor binds to the cell, with the cell receptor, the LDL is endocytosed, and an adapter molecule called adaptin binds to the tail of the receptor and attracts different molecules called clathrins, and these coat the membrane. Now, this effectively causes the endocytosis, and it causes a gulf in the cell membrane of the liver cell, forming a little vesicle, so a little like transport vehicle, and that little vesicle is called is internalized in the endosome. So after entering the cell, the vesicle uncoats and fuses with said endosome, and everything that's inside the little vesicle essentially gets released within that endosome, and that endosome binds with a lysosome and effectively releases the content and the cholesterol within the LDL cell, and all the excess LDL receptors and stuff end up getting recycled and sent back out. And this is essentially how you get cholesterol into the cell. So endocytosis, with the help of adaptin and clathrin, formation within the endosome forming with the lysosome essentially breaks down everything and releases the cholesterol esters, and the cholesterol is now within the cell. Great. Okay, now quickly moving on to proteins, and can someone tell me how much of this you actually covered? Or like, do you guys cover this much? Because I know we didn't. But if not, I'll try and cover it very quickly, because I don't think I have too much time. Not really too much? Okay, brilliant, because I don't know too much about it. So I'll just get it down into the basics and the overview. So amino acids are taken in by the diet or broken down from muscle, and these are catabolized by the liver. So amino acids are used to synthesize glucose and acetyl-CoA. Nitrogenous bases, nucleotides, bile, liver cells, blood plasma proteins, a bunch of stuff. As you can see on the right there's the structure of an amino acid. So you need to know about two things in regards to the catabolism and breakdown of amino acids. So you essentially have the removal and excretion of an amino group, which will then get recycled and excreted via the urea cycle, and then the oxidation of the carbon skeleton, which is left over, and the oxidation of that carbon skeleton is what forms energy. Okay, cool. So deamination is essentially when you have to remove the amino acid group. If you remember the structure of if you remember the structure of it, you have an amino acid, and then you have the rest of it, which is like a carbon skeleton. So you need to firstly remove the amino acid group, and the amino acid group, as you can see on the left there, is deaminated, or in the name, remove the amine, and this gets converted into alpha ketoglutarate and produces ammonium. This generates an NADH. Yeah, okay, hopefully you didn't cover it too much. Okay, yeah. All right, now in the urea cycle, so you have the deamination, you have the production of ammonia from the deaminating of the amino acids. So ammonium is toxic and needs to get converted to urea in the liver before it can get excreted by the kidneys. So long story short, you have ammonia gets converted into this thing called carbamol phosphate, which requires ATP, goes through a big cycle, ends up with urea, and the urea gets excreted by the kidneys. So the glucose-alanine cycle is essentially effectively gluconeogenesis, and is how glucose is created from alanine, and alanine is essentially an amino acid. So you have transamination, which happens in the muscle. So you have your muscle protein. This is in cases of, let's say, starvation, right? So you have your muscle protein, which is broken down into amino acids, forming ammonia, and you end up with glutamate. And through the magic of transamination, you end up converting pyruvate to alanine, and you convert glutamate to alpha ketoglutarate. And the alanine gets shipped into the blood, and that alanine goes into the liver. That alanine is deaminated, so this one has the ammonia, and you essentially chuck out the ammonia and reconvert it back to glutamate. And the ammonia is then removed by the urea cycle. And because you've deaminated it, you have alanine converted back to pyruvate, which can then be converted to glucose via gluconeogenesis in the liver. And that's essentially how you create glucose from protein, is through this long little process here. It's not too high yield. Don't worry too much about it. This part is a little bit more important, which is the most important macronutrient of alcohol. So you guys would have had, I think, an ICL/tutorial about this, but essentially the detoxification and breakdown of ethanol occurs in the liver, and ethanol uses NAD plus and essentially creates the intermediate acetaldehyde, and that's very toxic. So acetyl-CoA is produced, and which can enter the citric acid cycle or be converted to ketones or fatty acids. And the process of converting ethanol to acetaldehyde to acetate ends up producing a ton of NADH. So this leads to a low amount of NAD plus. So it slows down the citric acid cycle and leads to an increased production of fatty acids, as the acetyl-CoA can't undergo the Krebs cycle, and instead needs to be converted to something else, such as fatty acids, leading to high levels of blood and leading to a high amount of fat accumulation. This is why alcoholics have fatty livers, because of the creation of fatty acids. And also due to the NADH buildup by converting ethanol via alcohol dehydrogenase to acetaldehyde, and then acetaldehyde to acetate via aldehyde dehydrogenase, you end up with a buildup of NADH plus, which then favors the pyruvate undergoing anaerobic respiration, if you remember, which leads to a high amount of lactate production. And that leads to a decrease in pH, causing a lactic acidosis. And that is really bad. And that can cause hypoglycemia as well. An important note is that Asian flush, or the redness, is caused by acetaldehyde buildup. And this is usually Asian flush because people from Central Asia and South Asia more likely have a deficiency in the aldehyde or in the aldehyde dehydrogenase enzyme, or high levels of alcohol dehydrogenase, which means ethanol rapidly gets broken down into acetaldehyde, or acetaldehyde doesn't have anything breaking down, and that's toxic. So the MEOS pathway is a different pathway that is mostly important for chronic alcohol consumption, and it essentially uses a different enzyme. So you see there's a CYP450 enzyme and catalase, which converts ethanol into acetaldehyde. And this one uses NADPH instead of NAD. And this essentially prevents the buildup of NADH. But this can only happen in small quantities of time. And this is still really, this process ends up creating compounds called FAEEs, which destroy your liver and pancreas. But only you really need to know about this in terms of chronic alcohol consumption. Yeah. So this slide is effectively a summary of what alcohol does to the body. So as a buildup of NADH and lack of NAD+, you get reduction in the rate of glycolysis, reduction in the rate of citric acid cycle, reduction in gluconeogenesis causing hypoglycemia. Then you get a buildup of lactate and lactic acidosis. Then you build up acetyl-CoA, which then gets converted to fatty acids and leading to fatty livers and hyperlipidemia. And there we go. As well, I mentioned earlier about chronic alcohol consumption and thiamine deficiency, and the thiamine is obviously really important for glycolysis and stuff like that. Important note, bicarbonate is given to drinkers often to try and buffer the lactic acidosis which occurs. Yeah, okay, I'll try and skip past that. Okay, so now I think onto the last thing, which is a great metabolic race. So these are the two really important diagrams, which I'll just go through quite quickly. This is really high yield, which you wouldn't really think for something which was so short. But essentially, the great metabolic race refers to the energy sources used by an individual at different time intervals. So in this case, they use during a run, and the graphic depicts the amount of energy supplied by carbohydrates versus fats throughout the endeavor. So they use the example of running a marathon, and having actually run a marathon myself, I will tell you one thing is that the amount of energy supplied doesn't really matter because you feel really fucking tired at the end anyways. So regarding your body's different fuel sources, so at rest, fatty acids are your main energy source along with ketones from your liver. Then in the short first burst of while you're running, you're mostly relying on your ATP reserves, a bit of anaerobic respiration, and creatine phosphate. So this is rapidly generating ATP in the first few minutes. So when they go into quote unquote moderate activity, which they classify as 30 to 120 seconds, you're using most of your free glucose in your blood, and that's using anaerobic respiration to break it down. Then in the first 2 minutes, you have mostly fatty acids, if you can see here. It starts off with mostly fatty acids, and that slightly decreases, and you get an increased use of your carbohydrates up until 5 minutes. An important note is that this doesn't mean that the amount of energy being used, quote unquote, decreases. It's got to do more with the proportion of energy. So while you're still using a lot of energy, the relative proportion of fats drops and then rises, and then for carbohydrates rises and then drops. So remember, it's proportion, not an absolute value. Then you have extreme activity. 5 to 30 minutes. So at about 5 minutes, you have 85% of your energy from carbs and 15% from fats. And this is when your stored glycogen and blood glucose is still being used. Here you have muscle glycogen converted to lactate, and at 30 minutes exactly, that's what they say, approximately 50% of your energy is from fats and 50% from your carbs. So that's an important note. After about 30 acids, the rate of fatty acid metabolism exceeds the rate of glucose metabolisms, and you have mostly fatty acids. Another important note is that after 8 hours of without eating, so let's say you're starving, your proteins end up being your primary source of energy. So you end up with muscle breakdown, and through the use of glucose-alanine cycle, you end up with the formation of glucose and gluconeogenesis. An important note is that while you're sprinting and for short bursts of energy, you use creatine phosphate. So that's very small dose or very small supply. So this is the source of blood glucose at different times. So immediately following a meal, you have exogenous or dietary glucose. Then 3 to 8 hours after a meal, you have the breakdown of glycogen in your liver and your muscles. And that eventually gets overtaken by gluconeogenesis after about 8 hours post-meal. And after that, it's mostly gluconeogenesis, and that's when you're starving. Okay, and I think, is this the last one? Okay, yeah, this is the last slide, I promise. Then you have the different tissues and their sources. So cardiac muscle mostly uses fatty acids from bloodstream, and it can't store them. And whenever it can't use the phase, it'll use glucose and ketones. And this only really uses aerobic respiration. The brain primarily uses blood glucose and is not able to store it, and it can't use free fatty acids. The brain will, when blood glucose is low, will use ketones instead. Yeah, so main point of maintaining serum blood glucose is to provide energy to the brain because it can only really use blood glucose. Red blood cells, because they don't have organelles, use anaerobic respiration, and they use mostly blood glucose. Then in skeletal muscle cells, while at rest, they're using fatty acids, and when there's a burst of activity, is creatine phosphate, and then motor activity is the glycogen stores in glucose, and then fatty acid and ketones. And then as the activity gets sustained, it's using triacylglycerides and F phase. And then while starving, it'll break down the protein to provide amino acids and then gluconeogenesis and stuff like that within the liver. So then you see on the right there a little table for everything that's going on. Okay, I'm sorry that I went a little bit over. But if you have any questions, feel free to ask them in the chat right now or message me. These slides are obviously uploaded. Then if that's it, I'll say we'll come back at about 12:30, if that's okay, because I think that's when your next lecture will be up. So thanks for listening, and I'll see you guys in a bit then. Okay, great. Cool. I'll just pause the recording and be back at 12:30.
Hello, everyone. So my name is Jade Guitera, and I'll be giving you a SWOT back lecture on theme 2. So that's HEP, HKS, psychiatry, and a tiny bit of ethics and law. We'll wait like 1 or 2 other minutes before I share my screen and start, rapid fire spitting at you, because there is a lot to cover today. This is an entire theme that has been condensed into 1 hour. So I'm really sorry if I speak too quickly. Don't hesitate to interrupt me, ask questions. This is something that's supposed to help you. So let me know the best way that I can do this. And I'm guessing a lot of people will see this recording afterwards as well. So don't hesitate. I have put my auth key onto the slide, so you'll have my student identifier, and you can send me an email anytime with that. You just add like @student.monash.edu afterwards. So even if this is like a year later and you're seeing this recording and something doesn't make sense, or you spot a mistake, or you just need somebody to check up with, don't hesitate to send me a message. Okay, I might as well start because we have a lot to get through. Once again, I'm really sorry. I'll probably put a timer on myself so I don't start rambling, because a lot of these subjects take a while to get through. Okay, let me try and share my screen. I'm not very tech savvy, so here we go. Okay. We are now sharing, and I will present. So yeah, welcome everyone to the SWOT VAC lecture. This sem has not much pressure because it's been a little bit odd. Doing this by Zooms are a little bit odd as well. This is my first Zoom lecture. Please be patient with me. But once again, if you've got any questions, interrupt me or send me a message afterwards. And I do have to give a special thanks to, well, year one lecturers, Kiss Panda and Dana Bowden, who did this last year, because they have helped me remember all that we did last year, which disappears from your mind surprisingly quickly. So let's start with HEP. Why should we actually care about HEP? One, it's easy points on your exams, and it's things that you shouldn't just forget until the last minute. Yes, less to know by heart than metabolism, so use those points and actually like know the base concept, know your acronyms. It's easy to do this. But also, this is stuff that will actually be useful the rest of your life. I bet by third year we'll have forgotten everything about the Krebs cycle, but how to deal with stress, and same with HKS and with psychiatry, are things you'll actually see in your patients every single day, and things that actually do matter quite a lot. So please actually listen for this. So first, let's cover integrative medicine. Once again, just interrupt me if there's anything you don't understand. Integrative medicine, this is the best practice combination of conventional and surgical treatments, or with lifestyle interventions and complementary therapy, evidence-based ones. So this requires a holistic approach, viewing your patient as a whole and not only seeing them as a symptom or a disease. When we talk about complementary therapies, I'll be using the acronym CAM, that's complementary and alternative medicine. They're two different things. Complementary is something you use with a therapy to enhance outcome, minimize side effects, reduce costs, reduce symptoms. Alternative, it's used instead of the conventional treatment. That you need to be a lot more careful with because some are reasonable, but some can be unsafe, and you can't just replace medication without checking first if it's evidence-based. So a bit more about this, just some stats to know. Okay, so in HKS, in HEP exams, you'll have two types of questions. You'll have the really straightforward questions where anybody who's not a sociopath can understand. And then you'll also have some really picky questions with some statistics or some things that Craig Hassett has mentioned once in his lecture. So don't freak out about those ones, but there's a few stats that's really nice to know so that you don't feel completely lost. So for complementary and alternative medicine, the stats to know, 60-65% of Australians use some form of CAM, and 72% do not inform their doctor when using complementary therapies. So that means to integrate CAM, we need to be informed, we need to communicate with the patient, because if they use this behind our backs as doctors, that can actually be really complicated to deal with. So the pros of CAMs, we've been through this before. It's mainly increasing outcomes, reducing side effects and costs. A lot of people don't actually trust Western medicine like we're taught in med school. And so you do need to adapt of what they believe and what they think is best for their body. But yeah, remain clear-headed about it all. Different types of CAMs, we've got the whole body medical systems, biologically based, mind-body based, manipulative and body manipulation based, and energy therapy. Also, yeah, be a little bit more touchy on that last one, less proof, that's evidence-based. So it's nice to know one or two examples for all of these so you're able to classify all your different CAMs and what type they are. Okay, now let's go on to essence. Yeah, you know the acronym, education, stress management, spirituality, exercise, nutrition, community and connectedness, and environment. Know this by heart. This is the, yeah, you'll do it every social history, and literally all of what we're going to see now is just breaking down this essence. So all of HEP is in this one word. Do learn the acronym and yeah, do it well. Let's start with education. So when we're talking about education, this is not like, you know, what you did at school or in university, though sometimes that can impact. You talk differently to a 14-year-old high school student than you do to a university professor. But here we're talking mainly about health literacy. So what do you know about health? Do you know evidence-based? Do you know how your body works? We're also talking about education about your own mind, habits and stresses. You can have somebody who's brilliant scientist and know all about health, but has no idea that their coping mechanisms are terrible for them. And in education, we're also talking about the capacity to achieve goals and to change behaviors. So this is all really linked to prevention, because without the education, we can't have prevention. And well, we'll go a little bit through this as well with psychiatry, but there's 3 different levels of prevention. Your primary prevention is the causes of the causes. It affects healthy people. So example for that would be your quit smoking campaigns. We're trying to stop people from having the first stages. Secondary prevention is in an asymptomatic person with a disease process underway. So all of your screening programs fit into secondary prevention. Tertiary prevention is trying to prevent complications of an already established disease. So these are things where, you know, if you've got chronic diabetes, or you're trying to prevent complications that can happen because of that disease process. Hopefully that makes sense. Once again, interrupt me if it doesn't. Little acronyms, we love them. Also, knowing them by heart, it really helps. So BASC is how to provide health education. So you look at the behavior, what people do, the attitudes, what they think, and then how you can improve that is through skills. You teach them things, and knowledge. For knowledge, we're talking about what people understand. So it's what we say, but it's also how they interpret it. And it's really important when you provide health education to remember that. ADAPT, awareness, decision, effort, perseverance, tolerance, and discomfort, is about how to change habits. I don't know how your year has gone, but we had last year a tutorial in HEP about making SMART goals for ourselves and going through all this. So hopefully you've had a little play around with this already, but it can be really useful when you're trying to incite change in the patients to just think about those, and also in your exams, they're likely to come up. SMART goals, okay, I can't stand that word anymore. Everybody has talked to me about SMART goals so much ever since I've come to university, but realistically they do work, and it's a really good framework to think of things. So let's go through them in a little bit more detail. When you're setting a goal, you want things to be specific, so not some vague example, I'm going to lose weight. You want to be like, I'm going to lose 5 kilos. And it keeps you accountable as well. Measurable, same idea, give it numbers, keep yourself accountable. Attractive, you have to make sure your goal is worth the effort. You know, it might be like, oh, this would be good for me, but if it doesn't motivate you enough to go through the pain and the discomfort of things, you're not going to do it. Realistic, yeah, we've all done that, where we're like, yes, I'm totally going to do this, and then you don't, because it's not realistic at all. So make sure the goals are realistic and timely, having a set date to go do things by, because otherwise you're not going to. And then I put up the Prochaska de Clemente cycle of change. Same, you go back a little bit in MUM on it. But cute little cycle where most of the time it's quite easy to place people on. If they're pre-contemplation, they have the problem, but they don't realize they have it. Contemplation, it's a lot about self-awareness and realizing you do have an issue. And then you plan about how you're going to get through things, you act, and then you have to keep on going with that maintenance, or you relapse. They had the cute little sentence of like, relapse isn't failure, but an opportunity to find a way to maintenance. Yeah, if that gives you hope, great. But yeah, we're all on the cycle and we're trying again and again. Stress, let's go there. So you won't be facing too much of it this semester, hopefully. But yeah, you're going to face it all through your medical school, medical career, and I'm sure you've been through this through high school as well. But there's 2 different things. There's anxiety and stress. Anxiety is being worried about a future event. It's often disproportionate, but it can be very valid. Stress is a perceived inability to cope with a present or future event. Keep that word perceived in mind. Stress is always subjective. So why do we care about stress? 1, we actually need it to survive. This is fight or flight syndrome. We've built this stress inside us because we used to have to run away from wild animals, but also because of this stress performance curve. So if you have no stress at all, you're not going to do anything, you're not going to have any motivation. The peak performance is actually when you have some stress that lifts you out of that absence of motivation and engages you with the task itself. And I've put a little overview of what the autonomic nervous system does down there, so do have a go look through it again. We don't really have time to cover it right now. But yes, we can see on the other end of the curve that high stress is not good. it can be harmful over an extended period of time. We'll go through this in the next slides. But also stress is important because we've all felt it. Your patients will feel it, you'll feel it, you need to know how to deal with it. And it's been increasing in our society. We're all more stressed. It's actually predicted to be Australia's future largest burden of illness. So an important thing. Let's have a little bit more a look into it then. So the effects of stress, it impacts your genetic expression, so your cell repair capacity, shortening of telomeres. We're going to go through telomeres a few times. I'm sure Craig Hastert has mentioned them thousands of times in his lectures already. So I'm not going to go too much into depth. But yes, it does shorten your telomeres, which is bad long-term for your health, and that's pretty much how you die. Stress can decrease your immune function. It can cause lethargy, tiredness, or headaches, nausea, decreased concentration. Emotionally, it can make you depressed, or it can make you angry, eventually lead to burnout. We'll go for burnout afterwards. And it's also an independent risk factor for some diseases. There's also a high allostatic load on your body. So that means the wear and tear of your body. Your body is functioning too hard, too fast. And so with a prolonged activation of your autonomic nervous system, the one used in fight or flight, it'll cause a bunch of problems in vascularly, with atherosclerosis in your bones, with dysregulation of your immune system, of your endocrine system, with your hormones, causes central obesity, high blood pressure, high cortisol. The list goes on and on and on. The problem with most things in HEP is that everything is connected. So it's hard to give an exhaustive list of what it does. But too much stress for too long, not good. And it leads to burnout. So signs of burnout is lack of personal achievement. You're just emotionally exhausted. You don't feel like yourself anymore. And burnout, because Craig Hastert puts burnout as a sign of burnout. Look, this is something to look out for mainly for yourself and for your friends. Like, yes, learn it for the exams, but this is very likely in medical school. 28% of medical students report having burnout by their final year, and 75% of interns have burnout after 8 months. I've been through this before. Your friends will go through this, so do look out for this. It's serious. How can we deal with it then? Stress management, with the benefits of pretty much all of the opposite of what the negatives have been through, with like also an improved sensitivity towards others and increased empathy, and some positive coping skills, more feelings of connections. So how do we get there? Through mindfulness. So that can be through meditation and actually like formal activities, sitting down, controlling stress, but also it can be daily activities, you know, going for your run, connecting with friends. It can be for organization and goal setting, but it can also be just a mental practice of accepting, let it go. Yes, this is stressful. I can't control this, but it'll be okay. So the mindful revolution, yes, we take ourselves very seriously. So let's have a little look at attention. There's different types of attention. You can have the wandering mind, which is not necessarily focused on the task at hand. It allows for thought and creativity, but it can cause a toll on your happiness and your concentration if it's used too much. And it leads to the default brain. So what Craig Hastert calls the default brain is the inattentive mind. You lose capacity to distinguish between what's the imagination and what's the present. And yeah, it causes things like depression, anxiety, attention deficit, because you're not actually focused on what's happening right now. You're caught in the past, you're caught in the future. And that's something that happens a lot when you're multitasking or task switching, not focusing on what's happening right now. So being mindful is the solution to all that. It's being present to yourself and the world. There's different steps in achieving that. And I'm sure you've been, hope you've been to your tutorials about mindfulness and meditation. But one of the ways to do things is regulating your attention. So you know where your attention is and you bring it back to what you're doing. So you have control over that attention. And a mindful attitude of openness, of curiosity, acceptance, and self-compassion. All of that is things that you can adapt to your everyday life quite easily. So what are the benefits of mindfulness? You have better performance, it slows your aging, you're more emotionally intelligent, it helps your mental health, there's less medical errors, better health outcomes. It's a wonderful, wonderful thing that will solve all your problems, apparently. But do give it a try. Okay, on to the next S. I have to hurry up a little. I'm sorry if I'm already speaking so fast. There's just so much to So spirituality, that's a relationship to meaning, how we express it, where we find it, and how to look for it. This can be through religion, like faiths and belief, but it can also be through philosophy of what science, what is knowledge. It can be through altruism of giving to others and service. It can be creative, artistic, or it can be about your relationship to the world itself outside of you. So a lot of environmentalism and taking care of the world outside. There's many different ways to have a spiritual connection. Often you'll have more than one. Quite interesting to have a little thought about yourself and what spirituality means to you. Key stat is that 83% of patients want a doctor to talk about their spiritual beliefs. So now you'll have to do that every single history you take, at least for all of your OSCEs. And it is something that can help patients a lot when they're going through a tough time to have a relationship to what they believe in. So it's something to keep in mind. Benefits of that, it influences decision-making and treatment, impacts health, whole list of things, and impacts your coping strategies. We'll go through coping a bit later in psychiatry. You can have some issues around spirituality that you need to keep in mind. Some people have really negative experiences with religion or with stigma around it. So they have a negative religious coping. And certain extreme faith healing or beliefs in sex can also be dangerous for the person. So not all positive. I've also put here the two views of happiness, hedonic, which is about pleasure and avoiding pain, or eudaimonic. These are ancient Greek words. So this has been around for a while. And that's happiness being about meaning and personal growth. And usually these people do better in life and have a better health system. So also an interesting view of what is happiness. Exercise. This is a bit of a marathon in itself, honestly, this lecture. So benefits, reduce mortality and chronic diseases, a delayed onset of a whole different range of things, diabetes, obesity, and also it slows your degradation of telomeres, so must be amazing. No, realistically though, exercise is one of the key health factors that is protective for a whole range of disease. There's different types. Aerobics, that's your cardio, it's the best for your body. There's resistance, which is weight and strength training. It's also good, but you can easily go over the top with that one. And balance and flexibility, it's also important. It's mainly for people that can't do the other two. So little stats about exercise in the population. Males tend to have it do more than females, and there's a dramatic decline after 18 years old, where people just stop exercising, with the lowest being at 45 to 54 years old, often because they just don't have time to anymore. It's not a priority. Most likely to be inactive are women, people with a lower educational socioeconomic status, parents with children under 5, people speaking other languages than English at home, so important cultural factors. The reality is that 70% of Australians over 15 are classified as sedentary, with certain barriers to exercise as lack of time, motivation, or money, certain physical injuries or conditions, and also the convenience, like can you exercise, where will you do it? So we don't really reach those recommendations. You can have a look through those. That's what is considered the optimal level of exercise, because too much can also be bad for you. But yes, we don't really reach those anytime anyway. But very important exercise to keep in mind as risk factors. Nutrition, other really, really important risk factor for a lot of things. Key statistics, you can have a read through those. Dietary guidelines, I'm sure you've heard about the Mediterranean diet a few times now, but that's usually with whole fresh foods, mostly plant-based, very few empty calories, so very few fast food or added sugar, salts, fats. You need to have things that are varied and well prepared. Just having a salad every day will not help if you don't have anything else to complement it. And water is important. A little look on telomeres, once again, you can count the number of times I've said that in HEP already. So things that lengthen them are good for you, vitamins, folate, omega-3, things that shorten them are bad for you, processed meat, smoking, bunch of stuff. So yeah, that's a little overview of nutrition. I'm sure that you've talked more about in your tutorials, is things to keep in the back of your mind, and mainly in second year you will have whole module about nutrients and macronutrients. So you'll have a lot more information about this. This is just your first taste of it. Connectedness, we're almost there, guys. So also really important, and especially in COVID right now, we've had a whole lot more attention about what is isolation. So isolation is a lack of connectedness, feeling excluded and being socially withdrawn. It's different to solitude, which is being physically alone, but without all the other characteristics of isolation. So that can be voluntary, it can be healthy. You can be alone and not isolated. But social isolation, feeling alone, you can be in a room full of people and still feel alone. That tends to double death rate, and that was pre-COVID. So I'd be quite interested to see the stats now. We've learned to deal also a lot better probably with isolation. There are some inherited factors, inherited fears and intergenerational stress around connectedness. And we have also few interesting studies about brain development in children that have been abandoned. As a child, if you do not have social connection, you cannot develop. So this is actually really important and something that's harder to measure than other stuff. So important to keep a lookout for. Source of the connectedness, marriages, unless unhappy, contact with family, friends, religious groups or affiliations, being a member of something, but also mindfulness and happiness tend to help your connectedness. So this will also, you'll also ask in your social history, in your clinical skills quite a bit, because if your patient is connected or not will have great impact on them. Last one, the HEP, environment. So a key stat, 1/4 of all global disease is due to modifiable environmental factors. So the 3 components of environment, you've got the physical stuff, the air, water, sound, space, the human-made, town planning, urban, architecture, that's also really important, and the non-physical. So your domestic environment, your social environment, your cultural environment, work environment, study environment, this has so many impacts on your health, and you start realizing them more and more as you go through med school. But direct stuff like your air quality, chemical exposure, the climate, heat, but also indirect to your mental health, to your community, to your living conditions. So environment is actually quite a big deal, and climate change is gonna hit us like a truck. So we'll have changes in so much things, in your heat, in your weather, in the quality of water, the availability of space. So yeah, this will be one of the biggest challenges coming, just you wait. So environment is not to be forgotten. Whew, we made it through HEP, guys, congratulations. We're gonna go on to HKS now. Once again, if you've got any questions, please let me know, send me an email, but we need to move on. So health knowledge and societies, quite a lot of marks, so do have a look at it. This also includes med law and ethics, but also, once again, this is key to whatever patient you're going through. You see, they've been through social determinants of health. So what is health? A state of complete physical, mental, and social well-being, not merely the absence of disease or infirmity. Would recommend knowing that one off the top of your head. It'll make you sound smart, even if you don't really know what you're talking about. But it's also the goal of what we're all achieving. I say goal because realistically, none of us are completely physically, mentally, and socially well. And well-being itself is a subjective concept. So we don't really achieve this anytime, but it's something we're striving towards. We can measure health through quite a few different things: life expectancy at birth, the disability adjusted life years, DALYs, you'll hear quite a lot about. That's the years of life lost, and the years of life lost in disability of diseases. Maybe it's a good idea to have a look at how they're calculated, familiarize yourself with them. We don't have time to do that right now, but you will see a lot more of them, and incidence and prevalence too, in epidemiology, EPI, which is what replaces HKS in next semester. So good luck with that. But if you don't know about this right now, don't freak out. You'll see a lot more of it. Primary healthcare. That's also an ultimate goal. It's reducing inequity in health, considering it as a fundamental right. So how do we achieve this? We need a healthcare that is evidence-based, which is practical. It's culturally and socially acceptable. It's accessible regardless of your status, your wealth, your ability. It includes education, so you can be self-reliant, and it's participatory. So the individuals and the communities can self-determine what they need and what's going through. The elements of it, you've got also a nice little acronym. Would recommend knowing these, just because the way HKS works is in exams is usually with short answer questions. And so there will be keywords that people are looking out for, And if you know your acronyms and you can place your keywords in, you'll get full marks. These are often quite tick boxes. So even if you know the concept, but you don't know the words that you need to know and your concepts, it'll be a lot harder. So determinants of health, those are biological, behavioral, environmental, and social. We've been through environmental a bit with HEP, what we just did, so I'm not going to go over them. So let's look at biological and behavioral. Biological is what's related to your body and its biology. Some things you can't change, your genetic position, your age, sex. We're talking here about sex, not gender. You can decide any day to change your gender. Sex is a bit harder to change, but I wouldn't say it's still considered non-changeable, but this is, yeah, we're working on it right now, and hopefully in the future, people that don't agree with the sex they're born with will be able to change it. So yeah, this is in movement as we speak. Some things that you can change is like your body weight, your blood pressure, your cholesterol levels, and that's often linked to your behavioral determinants. So that's the habits and things you do: exercise, diet, tobacco smoking, alcohol consumption, sun protection, but also what you choose to access within health and screening programs. There's a lot of that, and those depend on the social determinants of health. So those are the causes of the causes, and those can be modifiable, non-modifiable as well. They can be positive factors or negative depending on where you're living and how you're living, and they can have proximal, intermediate, or distal effects on health. There's so many of those, we're not going to be able to go through them. I'm sure you remember, if you have my 3 who teaches you as well, he talks about ontological multiplicity. It's how everything is literally tied up and you can't separate one from the other. So a few examples are income, socioeconomic status, education and employment, culture, geographic location, if you're rural or not, which country you're living, developing, developed, gender and ethnicity-based social status, racial status, we're hearing a lot about that at the moment, lifestyle choices. There's a lot of those. A good way to try and like categorize them in your brain is with the 4 S's: stuff, space, and systems. So yeah, hopefully that can help and make things more logical in your mind. Some important concepts we need to know is inequality versus inequity. Okay, equality is the same treatment for everyone, and equity is a treatment based on need to achieve equal positive outcomes, and you need positive discrimination for that. So I think the picture kind of says it all. If you give the same thing to everybody, not everybody is going to have the same outcomes. If you give more to the people who need more, everybody can achieve that equal status. But we need to achieve that equity, we need to be aware of our implicit biases. We've got attitudes, common paradigms that have been told to us by society and our families forever. So they're going to affect our understandings. Even if you don't think they do, we are all full of implicit biases. And structural violence, there's a hegemonic discourse, big fancy word to say there is one way of seeing things that dominates, and people that don't really fit into that, what tends to be white, male, cisgendered, of a higher socioeconomic status, if you don't fit in there, minorities tend to be discriminated against. And it's something that we have to acknowledge because it impacts this so much, our access to healthcare. So I put the picture of the Tanahashi framework, and we'll go through the steps of that a little bit. The first step is availability, whether something exists or not. You can see that quite a bit in developing nations or rural. If you literally don't have the infrastructure, there's no way you can access it. Accessibility though, even if the structure is there, can you access it? Is it easy to access, but also easy to use? And that relies also on acceptability. Is it culturally acceptable? Will you be stigmatized and discriminated against if you use it? And that's how we get to actual effectiveness of our healthcare. You need to have all of those steps to get there. And that's, there is a lot of people that are at risk of poor access, including Indigenous Australians, refugees, people with disabilities, people living in rural and remote areas. So we're going to have a look at a few of those, and you'll have seen them in your tutorials as well. But it's important to keep an eye out for that. And I've also put the 2 types of equities up there. So Indigenous health, there is a clear health gap, and I know quite a few of my friends who are international students had never heard about this before, but if you're studying to be a medical doctor in Australia, if you're going to be working, at least as a medical student in hospitals, you need to be aware of this. There is a clear health gap with Indigenous populations tending to have higher rates of diabetes, asthma, all of these different chronic diseases, infectious diseases. There tends to be, that relies also on the social gap that we have. Indigenous populations tend, this is not all, this is just a general tendency, they tend to be even lower socioeconomic status with less employment abilities, poorer housing, poorer access to education and health and healthcare, and even a discrimination within the healthcare. Now, this isn't because just Aboriginal and Torres Strait Islander tend to get sicker. It's because of all of these historic causes. In case you hadn't realized yet, the white men came here over 200 years ago and stole land, spread disease, we had frontier violence, they segregated populations with, you know, massacres, and then there was the idea of eugenics, that we had a white race that was somehow so much better than everybody else, and everybody else had to disappear. And so there was a whole stolen generation as Indigenous cultures, Indigenous childs were stolen away from their families to be incorporated in white families and brought up as white with the idea of breeding out black in them. Even saying it seems crazy. And so somehow all of this has created a health gap. Who would have guessed? And the Aboriginal health gap has been recognized in 1960s. We've made some rules, some regulations, we've like, you know, assigned some healthcare resources to it. Unfortunately, not much has changed, not much, you know, there's a lot of talk now and a lot of intentions, but realistically, the health gap is still there and we need to keep working on it. So please do actually pay attention in your Indigenous modules that you'll have, because this is a complex societal problem and we need to face it. We have a responsibility as doctors. A few different stats and characteristics. 2.5% of the Australian population is Indigenous, and the criteria to be Aboriginal or Torres Strait Islander is to be of that descent, to identify as Aboriginal or Torres Strait Islander, and to be accepted within the community. That's it. It's not about like how many of your ancestors are Indigenous. It's not about the color of your skin. That is literally the only criteria. And the only way we're going to be able to achieve change is if we are aware of cultural blindness and cultural safety. So cultural blindness is a tendency to treat everyone the same and not realize that there is different cultures that often leads to miscommunication and institutionalized racism. What we need instead is a cultural safety, respect for cultural and social difference, and actually making change. I'm sure you see there's a lot of talk right now in the world about race and about racism and how we're going to, as a society, go forth from that. So no matter your political beliefs, if you want to be a doctor, you need to be aware of those two things, and you'll go through it a lot more. Refugee and asylum seeker health. There's a difference between the two. Refugee is a person forced to leave their country to escape war and persecution, natural disaster. And asylum seeker is a refugee that has applied for the refuge but hasn't been granted it yet. And so there's many different health issues linked to that. There's mental health with stress, depression, anxiety, post-traumatic stress disorders. Often they've lived through very traumatic things, and there's the lack of connection that we talked about in HEP. There's some infectious diseases that purely from being in another country you're a lot more exposed to, and difference in vaccinations that you can have had. There's certain nutritional deficiencies. There is a lot of characteristics, and it's something that we also need to be very aware of like the history of the person and where they come from. Yeah, rural health, I'll go through this really briefly because you'll have a rural health week next semester. So you'll see this a lot more in depth, and there's not much point for me ranting about it now. The general idea is that there's an undersupply of healthcare, rural GPs, but also just infrastructure and access to healthcare. So the standard of health is generally lower. You can use the 4 S's, staff, space, and systems model to think through this. And what, like, it's a lot of common sense of just where if you're missing certain things, you're not going to have an equal access to healthcare. So we are trying to do something about this. The AMA, Australian Medical Association, has tried to increase the proportion of rural medical students to 1/3, with the idea in the back of their mind that if you come from a rural place, you're more likely to go back to it. Though, as one of my rural friends has said, I left for a reason, I'm never going back. So I don't know how true that might be, but that's one of the steps they're taking. They're trying to use pharmacists for repeat prescriptions instead of traveling to GPs, and in general have more allied health worker that can take over from GPs that often have just too much of a population and geographical space to look after properly. And having more specialist vocational training places, because if you train rural, statistically you're not more likely to want to work rural afterwards. And lastly, a little overview of Australians' healthcare system. So we've got, yeah, you might be really familiar with this, but once again, you might be an international student and you haven't come across this before. So Australia has a mix between public and private system. So the private healthcare is if you choose to purchase health services that are not covered by the public system. And so we have this view that the private helps to fund the public. The people that can afford that extra healthcare make it possible for the people that can't afford it to also access the public. The public system mainly relies on Medicare. So this is a Commonwealth-funded universal healthcare insurance scheme. So the idea behind that is that the government pays for a basis, it pays for bulk bills, and doctors and certain procedures can ask if that doesn't cover them, they can ask for an extra, what we call gap, that will have to be out-of-pocket expenses, but Medicare covers that base. And it receives funding from the Medicare levy and general tax revenue. The PBS is the Pharmaceutical Benefit Scheme, so that's access to medication, which is subsidized. And the eligibility is the same as Medicare, Australian citizens, permanent residents, New Zealand citizens, and certain visas allow you also to access Medicare and the Pharmaceutical Benefit Scheme. So this is mainly a safety net, but if you use more in a year than the lowest dose, then the standard that is paid, you will also have to pay an extra gap on top of it. And concession holders have more of that safety net than others because they they're less able to pay and often have more health problems. So the actions of government, I've put it up there. My slides will be accessible on the drive. So do have a look if you want. You don't really have time, once again, to go through all of this, but do have a look. Certain issues with Australia's healthcare system. I'm going to touch very briefly on this. There are many, many, and I'm not aware of most of them. I think we'll just have to realize and deal with it as we go through medicine. One aspect is the pharmaceutical industry. One of the issues is that 30 to 35% of revenue is spent on marketing, which is twice the expense actually spent on the research and development of the medicine itself. It's true that there is this conflicting business goal between the business trying to make money and public health, which is just trying to make medication that is accessible to everybody. And that's inherent to the pharmaceutical industry. It doesn't mean that, we can't demonize big pharma and pharmaceutical companies. It's just you have to make sure that the practices that they have are ethical. And it's also your responsibility as a doctor and a medical student to make sure you aren't influenced and keep a level head about all this research and see what is evidence-based about treatments. Other issues with the healthcare system, it's an unsustainable system for certain reasons. We have an aging population, and so this aging population will have a lot more health issues as we go along. We have an increasing cost of pharmaceuticals and technologies, so people and the government will be less able to pay for them. We've got a certain lack of health professionals, and we've got not much of a focus on preventative health, which would be what saves us really, because investing in preventative health helps, not have to treat issues later. So this is only like a very brief overview. There are many other issues. There are also many really great positive sides to the Australian healthcare system. It's just about keeping yourself informed and keeping an objective eye on things. Okay, ethics and law. Sorry, I'm really parched by now. We're going to go through this quite quickly as well. And there's just certain principles. You'll have done your assignment on this already, so you'll know most of what we're going through. And this is often only a few questions or one short answer question, so it's concepts you need to know, but not necessarily in depth. Okay, certain ethical theories. Utilitarianism, having the greatest good for the greatest number of people. That tends to be the mentality in most governments, most government views, and making sure big hospitals trying to do the best for the greatest number of people. Another view of things is the right-based ethics, that all individuals are guaranteed certain institutional liberties and rights. So things like deserving to live and freedom and access to food and healthcare. Another view is virtue ethics, that a person who's virtuous will make the most reasonable decision. So that's kind of the idea of it's the intention that counts. If you're trying to do well, then it's the best. It's kind of opposed to consequentialism, which is not about what you're trying to do, but just about the result, the consequence. Is that right or wrong? So all of those are kind of general ethical theories, and that link into the principles of medical ethics. So those concepts, learn them by heart, understand their intricacies, understand how they work. I'm sure you've had a little taste of that through your assignment, but non-maleficents do no harm. Less straightforward than you think, because we've got side effects of medication. Surgeries, you always have to do harm. So it's really about the balance between non-maleficence and beneficence, trying to do good. Autonomy of patients, really important as well. And we'll have a little look at that through consent and confidentiality and making sure the person can make decisions about themselves. And justice is about trying to give equally and equitably to a lot of people. So we've been through that before as well. A little look on paternalism. Medical paternalism is overriding a patient's preference and autonomy, but it is believed to be in their best interests. There's two types of them. There's weak paternalism. It's justified when a doctor's actions align with the patient's intentions and when they lack the decisional capacity. So sometimes, you know, you have the best interests and they are not able to make a rational decision, so you have to step in. And that can be very positive. You just have to, once again, exert your common sense and what you've learned in ethics and decide what's right and wrong. Strong paternalism is always problematic. It's when the doctors actively override the informed, voluntary, competent decisions regarding treatment, and it breaches consent. Confidentiality is key to doctor-patient relationships because, well, you need to make sure that you don't, you're going to hear a lot of really personal things from your patient, and building that trust is so important. There are some reasons that you can disclose things if the patient consents, if the information is de-identified. It's really important for research. We need to know certain things. Information is being shared within a healthcare team. You're not working alone, so you do need to share some information. If there's a threat to public safety, or if the patient poses a threat to themselves, then you can disclose information. There's also certain mandatory reporting, such as notifiable disease and all the others that are listed down here. We're running short on time, so I'm not going to go through the list, but do know all the ins and outs of mandatory reporting because it is really important, so worth revising. Consent, so important within medicine, within general life. Just you need to make sure that the person consents voluntarily. It's not coerced, it's not influenced by other people. It's their own decision, that it's informed. They have to know all the risks, the side effects, the reasons for the procedure, what will happen if they don't have the procedure, the results of what could happen. It's really important, and they need to be capable. So they need to be of sound mind, so not under the influence of drugs or alcohol. They need to be over 18 or almost. There's like a little bit of leeway if, you know, you're 16 and very capable, then that's okay. If you're 16, you're not capable, yeah, that you need to keep that in mind. And that's also a lot of exerting your common sense and your values. So consent can be oral. Just said like, oh, is it okay if I do this now? You'll do this at the beginning of every examination, every history for the rest of your life. It can be written. That's mainly for big procedures, for things when you're not too sure if the person understands, so you make sure you write it out for them. And it can also be implied. This one is the trickiest one because because it's not set out so clearly, it can cause more confusion. So it can be during emergency situations, if it's a necessity, any reasonable person would consider it necessary, and it's often when the patient can't give consent. And it can also be if it's very routine daily procedures. You can't ask for consent absolutely every time that you do certain things. So if they've accepted once, it can be implied, and they show signs that they're okay with it, it can be implied. But once again, very tricky, and so please be careful with that one. Two ideas mainly for law for that one: battery and negligence. Battery is intentional contact without a patient's consent, but it's not necessary to be like an injury. It can just be like you touch them without asking them. Negligence is a breach of care resulting in unintended injury. So you need to have the duty of care to be present. You need the doctor to breach that duty of care. You need injury to result from that, and you need the injury to have been foreseeable. Basically, like you knew that this would happen and you still went through with it. That's negligence. It's not known in injury as like, surgery, you put a cut through there. It can also be like side effects of medication. If you're mixing two medications that you know shouldn't be mixed together, it can be considered as negligence. Yeah, I remember our tutor in med law telling us that, we'd be sued for sure every 10 years of our practice. So this is stuff that, like, all this ethics in law might not be so important right now, but it's stuff that we need to know and we need to be aware of, because we'll be faced with it. Medicine of the mind, last one. I don't have much time to go through this, so once again, I'm going to speed. Please ask questions and let me know. Also very important, and you will see more of it. So the biopsychosocial model, it's really important and ties back a lot to HEP and what we saw at the beginning. Is the idea that not as we thought before, your body is not a machine, it's not mechanistic, it's not purely either consequences of biomedical processes in your body. Freud discovered that your mind actually does quite a lot, your psychology, and now we're incorporating as well the social side of things. So biology, psychology in your mind, and social, all those social determinants of health. And if you want to actually treat your patient and not just his disease and symptoms, you have to incorporate all of that. So these can be internal factors or external factors, and they're also really impacted by behavior. So behavior being the actions and your reactions to a situation. And that's important for learning. You need to change behavior occurring as a result of prior experience. So we're going to have a little look at learning right now. An example for that is classical conditioning. Okay, so there is a lot of mind-boggling words because they all sound similar. Unconditioned stimulus, which is just a reflex response. Unconditioned response, it's your reflex. The unconditioned stimulus gives a reflex unconditioned response. The conditioned stimulus elicits a conditioned response after repeated pairings with unconditioned stimulus, and the conditioned response is the response now elicited by the conditioned stimulus. I feel like I'm rapping, just like making no sense at all. So let's go through an example. Has it ever happened to you guys before that like the song that you use as your alarm bell in like the morning to wake up suddenly comes on in a supermarket or whatever store, and your body just reacts and you're like, oh my God, I can't stand this song anymore? That's because you've trained your mind through classical conditioning. When you hear something that technically this song has no relation to whatsoever in your life, but you've trained yourself to wake up to that song, your body will respond every time. So you've got classical conditioning. You've paired an unconditioned stimulus with the conditioned stimulus to elicit a conditioned response similar to just waking up in the morning. Okay, I hope that makes sense. If it doesn't, I can go through it again at some other time. It is just try to think through it logically. It does make sense. It's just the words that are really tricky, but you need to have a mastery of those words. Operant conditioning, it's the consequences that immediately follow a behavior will determine whether they'll be repeated or not. So for that, you have reinforcement, consequences which increases the chance a behavior will be repeated, and punishment, the consequences decreases the chance the behavior will be repeated. So positive reinforcement is a pleasant stimulus added following a good action. I put good or bad to make things simple. It's not that clear. But for example, if you're given a child a lollipop after they do their homework, they're more likely to do it again. A negative reinforcement is a nauseous stimuli is avoided if you follow good behavior. So for example, if you put sunscreen, you're not going to get sunburned. Negative reinforcement. Your skin is fine, and so you're going to do it again. Punishment, positive punishment, it's an unpleasant stimuli added following a bad behavior. And negative punishment is a privilege or reward lost following bad behavior. So positive punishment give you a fine because you parked in the wrong space. You're non-likely to do it again, but you've been given something. Positive punishment can be a bit mind-boggling sometimes. Negative punishment is a privilege or reward lost following bad behavior. So if you take the example of the child, the parents are like, oh, you didn't do your homework on time, then you're not going to watch television or something. I've put a little table down there to try and make more sense in your head. Difference between those two: classical conditioning is involuntary, and the response is based to a stimulus from prior to the behavior. And operant conditioning is the response is voluntary, and it's as a consequence of the behavior. Other models of learning: superstition, I'm guessing you know what that is. Social cognitive theory, so that's Albert Bandura's theory of learning by observation and imitation. Classic example of that would be you watch a YouTube video about how to fix your bike, and then you know how to fix your bike. Technically, that's like a model of learning as well. Self-efficacy. So this is different to self-esteem. It's a belief about your own abilities and talents, but an ability to go through them, and it influences our behavior. So an example for that is, let's take your entry to medical school. You didn't necessarily think you'd make it into Monash, but you thought that you could apply to Monash. So that was self-efficacy. You were like, yes, I know I could. And if you didn't, you wouldn't be here right now. And so I bet you a lot of people never even thought it was a possibility and never applied, so never got in. So that's self-efficacy. If that doesn't make sense, then send me a letter, or like look at it again. An email. Did I just say letter? Attribution. So that's a process of inferring the causes of mental states behavior on yourself and others. So where you put the responsibility. They can be external, that's the demands of the environment. They can be internal, so reflecting the person's personality and abilities. They can be global, with a stability of reasoning, making excuses. Stable or unstable, they can be global, specific. Controllable versus uncontrollable. Remember, this is perception, so you think you can control, you think you can't control, doesn't mean they necessarily are. The certain times these attributions can bring what we call cognitive dissonance. Same, I'm putting a lot of keywords on there. If something doesn't make sense, please go look it up, because you need to know these. So this is a discrepancy, like a clash between attitudes or attitude and behavior that results in a state of psychological tension. So an example for that would be during COVID, my friend is sick, so I want to go visit them. That's what I usually do. But now I've been told if they're sick, I shouldn't visit them. And that can create cognitive dissonance where on top of being sad because your friend is sick, there's this clash of you don't know what to do anymore because two things that you've been taught, two attitudes, don't fit together anymore. Biases and errors of attribution. This is also quite important. Please do know it. Fundamental attribution is a tendency for observers to look at an actor and to underestimate the amount that external attribution is going on and overestimate the impact of an internal one. So for example, if he lost his job, it's because he's lazy, not because his boss is terrible. The actor-observer bias is kind of the opposite. It's the tendency for the actor, the person going through things, to favor external attributions and excuses for their own behavior, especially if it's negative. So if I failed, well, if we take the same example, it's actually this person has been really lazy and they're going to say, oh, it's my boss's fault, I've been fired. Self-serving biases. We all do these, and like once you've learned about them, you'll start spotting them out in other people. It's actually quite fun. But self-enhancing biases is attributing one's success to personal factors. I've aced this test, and it's because I've studied so much. A self-protecting bias, it's attributing one's failure to situational factors. So I've actually flunked this test, it's terrible, but it's because the teacher didn't teach me well, not because I didn't study enough. A self-handicapping bias is publicly making advanced external attributions for one's anticipated failure. So it's actually saying, like, I'm sure you've heard friends who've done it before yourself, where you're like, oh, I did absolutely terribly, just to make sure that if you actually do, you've like self-handicapped, and so everybody is kind of aware of it beforehand. The symptom perception and interpretation. We don't have much time, so I'll go through it really quickly. I haven't even put the common sense model because I knew we'd be running out of time, but do have a look at that one. It mainly goes through the idea that to recognize that you have a problem, you have to perceive the symptom. So a symptom is by definition subjective. It's a subjective evidence of a disease. So it relies on your attention, where you actually focusing on your body or not. It's affected by other things like external information, selective attention, negative effects. And then you need to interpret what's going on. So that can be influenced by many different things as well. And it will often cause an appraisal delay. So it's a time taken to interpret system that's influenced by denial. No way I have this, distraction, too much going on in my life. I can't focus on this right now. Minimalization, yeah, I have this headache, but it doesn't matter. Or misattribution, I have a headache, but it's because I've got a hangover, not because I've got a migraine. So being ill, that's quite linked to what we just saw with interpretation and perception of symptoms. You need to know what you have to seek help for it. So help-seeking behavior is problem-focused and trying to obtain external assistance for what's going there. You'll often have an illness delay, time between the recognition of illness and deciding to seek for help. So just to recap on that, you have to actually perceive your symptom, and then you have an appraisal delay where you actually realize that it's a problem, and then there's the illness delay where you're actually going to go seek for help. And the illness delay can change with the illness representations you have. So the timeline, the consequences, the possibility of the cure, and all that influences things. We don't really have time to have a full look at the sick rule of Parsons, but that's the idea that if you're sick, you're excused from your normal roles and responsibilities, you're not responsible for what's going on, but you want to get better and you will be cooperative with provided help. This sick role is not actually realistic to what's going on, and people often don't fit into that. So it's important to know that this is what most people think, but people will not actually act that way. And I put a few points on carers because it's also really important when you're looking for after a patient. There's a type of helpful caring and unhelpful caring, and it's important to analyze which category your carers are in. There's a lot of benefits to caring. You're feeling helpful, you have more connection, but there's also a lot of disadvantages of caring. Mainly, lack of time, somebody is depending on you, and so it's important to include the carers in your care of the patient. Stress, here we are again. There's different visions of stress. We've been through the idea of it as an internal process. I'm not going to go through that again. You can have it as an external stimulus or as a transaction between an external event going on and the individual reactions. That's kind of the mix between both. The stress can be acute, just happening like right now, right here. That can be something huge causing post-traumatic stress disorder, war or a natural disaster. Or it can be a routine, like that spike of stress when you're running for the bus because you're late or exams. But chronic stress that happens every day, can lead to burnout. And we've talked about that before. So stress as an external stimulus, basically there's a lot of theories around that, but it's not really true because it doesn't take into account the differences in people's ability to cope and the different perceptions of what is a stressful event. Remember, our definition of stress, the perceived inability to cope. So transactional stress actually makes a lot more sense because it considers the nature of the stress and the nature of the person experiencing it. So you have two appraisal effects for that. The first one is how the subject appraises the event. They can think it's really stressful or not. And then how they appraise their own resources and ability to cope. And the stress arises when the demands seems excessive compared to the resources the person has to cope. There's a lot of factors influencing that appraisal and whether the person will deem it stressful or not. And I put them down here. Impacts of stress, I've kind of gone through this before. They can be behavioral, organizational, cognitive, emotional, physical. and that can cause illness, or directly because your immune system shuts down or your hormones are firing away, but also indirectly because you're responding to stress by overworking yourself, not sleeping, smoking, drinking, eating. And a lot of things can actually modify our stress. Things like social support, where if you think you can actually control what's going on or not, and depression, anxiety, coping, a lot of things. So we've looked a little bit at coping already. You can do it well, adaptive, or not well, maladaptive. Your different coping styles can be problem focused, you change, you do something about it, can be emotional focused, you find support, comfort, or it can be attentional avoidant, that's not helpful. And yeah, I've rushed through it. It's been an hour already. Sorry for going through all of this so quick. So please, once again, let me know if there's something I can tell. I can also stay in this for a few more minutes if you want to contact me on like the Zoom chat right away while the next lecture starts. But I'm gonna have to let you go because you've got another lecture starting already, I think. So thank you so much for listening.
Thank you so much, Jade. That was really good. Sorry to have to like stretch you a bit for time, but it was still really well done, and you can always ask Jade anything. She's super nice, super friendly. I think next up we should have genetics, and I think Senara should be here.
Yeah.
Oh, she's here. Great. Okay, yeah.
Put my video on.
Yep. And whenever you wanna
Hello.
whenever you wanna share your screen, whenever it gets started, you can go ahead.
Yeah, sure. I'll just share my screen now. Can everyone see that? Yeah. So hopefully it's shared. I'll just present it, and I guess I'll start. So if these slides are quite similar, it's because I did this for the revision lecture. But I'll be going through some of the main concepts and what I think is quite high yield for the exams or the formative quiz, and we'll try to rush through so we can get to the main questions. So genetics, just gonna okay, there. So we're just gonna go through central dogma, DNA replication, transcription, translation, patterns of inheritance, genetic mutations, population genetics, and biotechnology. So central dogma. So essentially what central dogma is how your DNA is the genetic information from your DNA is turned to functional proteins. And that comes from DNA to RNA and then RNA to proteins. So DNA to RNA is called transcription, and the conversion of RNA to protein is translation. So just the basic structure of DNA. So the DNA composes of a deoxyribose sugar. So it composes of a deoxyribose sugar, a base, which can be adenine, thymine, cytosine, or guanine, and a phosphate group. And DNA is made, it's double-stranded, and it's in an antiparallel arrangement, which means one strand goes from 5' to 3', and the other strand goes from 3' to 5'. So DNA is really long, but it's kept in our body because it's tightly packed and stored as chromosomes, which is made of chromatin. And they're wound around these proteins called histone proteins. And histone proteins are formed from 8 smaller proteins called nucleosomes. And so then DNA and histone proteins are what makes chromatin. Okay, DNA replication. So the full process of DNA replication is semi-conservative. And what semi-conservative means is that the replicated DNA which is made consists of one new strand that's made and then one old strand. And that old strand is the template strand. And in eukaryotes, DNA replication has multiple origin sites where replication can begin. This just makes the whole process more efficient and fast. Multiple proteins assist in DNA replication. And I think this is a question the UMC really likes to ask. When does DNA replication occur? And this occurs in the cell S phase of the cell cycle. So I'm just gonna explain DNA replication using this diagram, and then you guys can read it on the next slide. So it begins with DNA helicase, which is a protein that essentially breaks the hydrogen bonds and unzips your DNA. So you can think of like a zip and how when you pull it down, it unzips. That's exactly what DNA helicase essentially does. And then you have topoisomerase, which allows your DNA to unwind and come straight. Because as you remember, DNA is in that helical structure, and topoisomerase just unwinds it. And since your DNA is very likely to want to go back to that structure, you have single-stranding binding proteins, or SSBs, that keep it straight and stable. So in the leading strand, as you can see, so to begin DNA replication, DNA polymerase just cannot start. It needs a starting point. So that starting point comes with RNA primase that puts a primer. And this allows DNA polymerase to attach onto the primer and then elongate and create new DNA. And that just adds nucleotides and moves on. So the leading strand is quite simple because DNA polymerase goes from 5' to 3'. And so it goes in the direction as DNA helicase is unzipping that DNA. But in the lagging strand, however, it goes in the opposite direction as it needs to go from 5' to 3', as you can see. So it has to go this way, while DNA helicase is moving in the opposite direction. So what happens is RNA primase puts down multiple RNA primers, and these are called your Okazaki fragments. So they're just tiny fragments. And then you have your DNA polymerase that attaches onto the RNA primers and keeps on elongating until it meets the next primer. And another DNA polymerase comes attached to the next primer and just keeps on moving like that. And then, and then you have RNase H, which is an enzyme that moves the RNA primers away, and then DNA polymerase 1 comes in and extends those DNA, replace the RNA with actual DNA. And then last of all, you have your DNA ligase that combines those fragments together. So that's all just summarized in this one slide. So everything is put in bold, so you can see the main points of what I'm trying to say with DNA replication. So that was a lot. If anyone has any questions about it, feel free to put in the chat, or I'll just move on to transcription. If anyone has no questions, I might just move on to transcription then. Oh wait, sorry. I think let's go do it DNA repair. So DNA repair has there's two mechanisms of it. Proofreading. Proofreading is a characteristic that DNA polymerase has. So what it does is, as DNA polymerase is elongating your DNA, if it sees a mismatched pair, it can look back, recognize that, and then remove it and put in the right nucleotide. And this happens during DNA replication. And then after DNA replication, you have another mechanism called mismatch pair, which is when a mismatch pair of bases is identified, and a whole section of DNA is just cut out, and it's replaced with a same section of DNA, but with the correct pairing sequence. And then DNA ligase comes in to seal the gaps of the phosphate backbone and creates the correct DNA sequence. So transcription. Transcription is how DNA is turned into mRNA. So just a brief summary of RNA. RNA is single-stranded. It is made of a ribose sugar instead of deoxyribose, and its bases are adenine, cytosine, guanine, and uracil. So it does not have thymine. And there's three types of RNA. So you have messenger RNA, which is the RNA that you can use to code amino acids. Then you have your transfer RNA that is used in translation, which brings amino acids to the ribosomes. And you have ribosomal RNA, which makes up your ribosomes. So transcription, there's just three phases of transcription. Your initiation, which is the beginning of transcription. So an RNA polymerase binds to a promoter sequence of a DNA, and DNA has promoter sequences on its strand for RNA polymerase to bind. And this binds alongside different transcription factors that promotes RNA polymerase. So this signals the DNA to unwind, and so that RNA polymerase can read the base pairings. And then you have step 2, which is elongation. That is just addition of the nucleotides to form the mRNA. And they use the same complementary base pairing. Instead, adenine is paired with uracil because remember there's no thymine in RNA. And just like DNA polymerase, RNA polymerase reads from 3' to 5', so it goes in the direction of 5' to 3'. And then you have termination, which is the ending of transcription. And this happens when RNA polymerase meets the stop sequence or termination sequence in the gene. And this signals the pre-mRNA form the strand to be complete and detached from the DNA. So this is just a picturesque view of it, of how you have the promoter sequences, RNA polymerase binds. This causes it to unwind, allowing RNA polymerase to read. And then once it meets a termination sequence, that's when transcription ends. And so we have pre-mRNA. And so pre-mRNA must be processed to make mature mRNA. And mature mRNA is made when a methyl G-cap, which is a sequence of guanine, is added at the 5' end, and a poly A tail, which is added at the 3' end. And this just ensures that the mRNA doesn't get destroyed in the cytoplasm. And then you have splicing. So splicing is when introns or the non-coding areas of your mRNA, which really code for nothing, is spliced out, and this leaves only the coding areas or the exons. And this is done via spliceosome. So a main aspect of splicing is that it can take all the introns out, but it can take different exons out as well to make so that one pre-mRNA can make different mature mRNAs, which in the end code for different proteins, which is just another efficient aspect of protein making. So now we have translation. So translation is when RNA is turned to protein. So RNA, so the beginning step is when tRNA has to be charged with the amino acids. And so this is just the binding of the tRNA to the amino acid, and this is done via the enzyme amino acyl tRNA synthase. And then the second step is initiation. So mRNA, the mature mRNA, binds to the small subunit of a ribosome. This initiates tRNA, which brings methionine to the start codon, and your start codon is always AUG. So tRNA brings the methionine, and then this signals a large subunit of the ribosome to form, and this makes the initiation complex. So now you have elongation, which is just the adding of amino acids to make the polypeptide according to the complementary codons. So I'm just going to explain using this diagram. So your large ribosome has 3 sites: an E site, a P site, and an A site. And you can figure out their function according to just their letter. So A is the arrival site, and this is when the tRNA arrives to the ribosome. And then you have the P site, which is the peptide bond formation. In the P site, one amino acid and the other amino acid forms a peptide bond in this P site. And then you have the E site, which is the exit site, and this is when the tRNA is free from its amino acid and it's just free to leave the ribosome. So in the elongation process, so you have your methionine from the A site, the tRNA from the A site moves to the P site, and the mRNA moves along the ribosome moves along and meets the next codon. This signals the tRNA with its complementary anticodon to bring the next amino acid. And then in the ribosome, they form peptide bond, and then the previous tRNA is free. This moves to the E site and it's free to leave. And this process just keeps on happening, like bringing amino acids, making a peptide bond, leaving. And this happens until the ribosome meets a stop codon. And a stop codon just initiates, just finishes the translation process. A release factor binds, causing the polypeptide to be released from the ribosome, and your protein is made. and that's illustrated in this diagram. And then you have post-translation modification, and this is just the modification of your proteins. So as you can know, proteins have different structures. You have primary, secondary, tertiary, and quaternary. So proteins need to fall into the three-dimensional structure, and this can be done this can be facilitated in chaperone proteins, which are essentially these proteins that have a cavity inside them, which allows the polypeptide to just go in that cavity, fall to its correct structure, and then come out. And this process requires energy. And different proteins can undergo many processes like glycosylation, which is just adding glucose, phosphorylation, adding phosphate, proteolysis. Some proteins require some degradation of their polypeptide, and then lipidation, adding lipids. So these are just different processes according to what the protein's function is. And this occurs in the rough endoplasmic reticulum. And if you're making excess protein, which can happen, these excess proteins are broken down by a protein called ubiquitinine. Can't really pronounce it. So yeah, that's all of central dogma. If you guys have any questions, feel free to put in the chat, or we can start with the questions. So okay. So question 1, you guys can answer. In which phase of the cell cycle does DNA replication occur? Just gonna give you a few seconds to answer. So DNA replication, as I said in previously, it does occur in the S phase of the cell cycle. Okay, your second question. If a DNA strand comprises of 30% of adenine, what percentage of cytosine does it contain? And just remember your base pairings of what bases pair with what. So yeah, a lot of y'all are answering 20%, which is correct. So I just put the calculation in here. So yeah, it's 20%. As you remember, adenine, percentage of adenine is equal to percentage of thymine. So that all equals up to 60%. So then if you minus that from 100, you get 40, and then you divide it by 2 because you need the equal amount of cytosine and guanine. Question 3, which enzyme matches to their correct function? RNA polymerase forms short RNA strands from DNA polymerase. Topoisomerase unzips DNA. DNA ligase joins DNA fragments together in the lagging strand. Or DNA helicase stabilizes DNA strands during DNA replication. Just gonna give you guys a few seconds. So yeah, 3 is, sorry. So yeah, 3 is the correct answer. So RNA polymerase, the first one is actually RNA primase. The second one is DNA helicase, and the third one's correct, while the fourth one is just your SSBs, just single-stranded binding proteins. So your next question, which of the following codes for methionine, lysine, tyrosine, valine sequence? This might take some time, so I'll give you as much time as you want to get through this. And this is a question that is quite representative of what you would get. So it seems that this one is taking quite a bit of time and there might be a bit of confusion. I'll just explain it. So you have your mature mRNA, and you need to look at the sequence of codons that meet, which is the same sequence as methionine, lysine, tyrosine, and valine. So essentially, you need to translate what the sequence is. So this is taking some time. I'll just move on to the answer and I'll explain it, if that's all right. Just tell me in the chat if it's a bit too hard and I'll just explain it. Okay, I'll go to the explanation. So you have your methionine, lysine, tyrosine, and valine. So using this amino acid table, as you can see, AUG is methionine. That I'll just go one by one. So your first one, AUG is methionine. So that's correct. Then you have lysine in your table that codes for AAA or AAG, which is again correct. So you can see AAA is here, the triplet codon. And then tyrosine. So the three options of tyrosine in this table is UAU or UAC. As you can see, this one, As you can see, this one is a methionine, which means it's wrong because the next one should be tyrosine, right? And the next two options are also wrong. As you can see, it's UUC, which is just different other amino acids. Your second one, just in these kinds of questions, if they start with methionine, immediately look for AUG because that's the only codon that that translates to methionine. So your second one, you immediately see AUG, and then your next one is lysine. So your two options are again AAA or AAG. So AAG, and then your next one is UAA. And UAA in this is a stop codon. So you can just immediately disregard that. And your third one is your correct answer because you can see AUG, which is methionine, and then you have lysine, which is AAA, and then tyrosine in this box of options is UAU, and then you have valine, which in this box of options is GUU. So third one is the correct one. This takes some time, and this is a question that you do get in some of your formative quiz. So in the purpose of this lecture, since it was quite time restrained, I understand. But does that explain it? Because I can go through it again if anyone wants. Feel free to just say it in the chat. If not, we'll move on to the fifth and last question. So if someone says, do you have to read it from 3' to 5', or does it matter? It doesn't really matter. You can read it from either end, but when you're given in this option, you generally read it just from left to right. It doesn't matter. Does that answer the question? Okay, no problem. So we'll just move on to the fifth one. This is also a long one. So yeah. So your fifth question is, again, another question that you might get in your formative quiz. Which of the following steps are in correct order in regards to DNA replication? Okay, so some people have answered D or B, and the correct answer is B. So the reason why it's not D is because, so B is 3, 1, 2, 4, 6, 5, 7. So 3, helicase opens up. 2, topomerase keeps it from, unwinds it, that keeps it from coiling back. Single-stranded binding proteins keep it stable. And then you have your primase that synthesizes the primers for the DNA strand to, for DNA polymerase to begin. And then DNA polymerase 3 extends the primers. The reason why it's not D, because I put 4, 5, I believe. Oh, I think, yeah, sorry. So B, yeah. So, yeah. So your DNA polymerase 3 extends the primers, and then you have 5, which your RNA primers are removed to replace it by DNA polymerase 1. Then 7, the gaps are sealed by ligase. Reason why it's not D is because your helicase works, and then your topoisomerase unwinds, and then your single binding proteins, the main function of it is to prevent it from unwinding, which means it needs to be unwound first. If that explains why it's not D. So good job. Now we'll move on to patterns of inheritance. So these are some terms that you guys will need to know and just understand what they are. Just for the purpose of time, I'm just gonna move on to the next slide, because, or I can just go through them very quickly. So a trait is a characteristic. A phenotype is the physical manifestation or the expression of a gene. And then you have a genotype, which is the genetic makeup of an individual, so what the gene sequence is. And then a gene is just a section of DNA that codes for a specific trait. Then you have your allele, which is the variant form of a gene. A locus, which is the location. Homozygote is an organism with 2 copies of the same allele, and a heterozygote is an organism with 2 different alleles of the same gene. So a human karyotype. A human karyotype is just a picture of chromosomes, and they're grouped in pairs and they're organized according to size, with your big ones from the beginning and your small ones later on. So humans have about 22 autosomal chromosome pairs and 1 sex chromosome pair. So males have 22 autosomal with an XY sex pair, and females have 22 autosomal with XX sex pair. And then you have your dominant, recessive, and codominant alleles. So dominant alleles are alleles that show that phenotype regardless of it being heterozygote or homozygous. And then you have your recessive traits that can only be shown when they're in homozygote, because when they're in the presence of a dominant gene, they can't express their phenotype. And then you have your codominance, is where both alleles are simultaneously expressed in homozygote, and the main one that comes to mind is your ABO blood group. So now autosomal inheritance. Autosomal inheritance is inheritance of a gene on an autosomal pair, and this means that males and females are equally likely to get that gene. Autosomal dominant is when a dominant gene is on an autosomal pair is inherited. So that's like Huntington disease. And in autosomal dominance, it won't skip generations. And then in autosomal recessive, it's when a recessive gene is found in an autosomal pair, but you need to inherit 2 of the mutated copies to get the disease, to get the mutation, or to inherit it. So one that comes to mind is cystic fibrosis. And in autosomal recessives, you can skip generations. It can happen, and this will create carriers. So people that do not show the gene or aren't affected by it, but have inherited 1 copy of it. So these are 2 pedigrees of what an autosomal recessive and autosomal dominant is. As you can see, in autosomal recessive, it really depends. It may not skip a generation, but if it does skip a generation, it's very unlikely that's an autosomal dominant inheritance. So now there's sex-linked inheritance. And sex-linked inheritance or sex-linked traits are associated with genes found on your sex chromosomes. And most of these are found on your X chromosome because they're bigger than the Y. And because of this, your men are more susceptible to sex-linked traits rather than women. Yeah, because they only inherit one X chromosome. So if they get it, they get the disease. So X-linked dominance, like the name suggests, is a dominant gene found in the X chromosome. And X-linked recessive is when a recessive gene is found on an X chromosome. And then you have your mitochondrial DNA. As you know, your mitochondria have their own small circular DNA. And hence, mitochondria and mitochondrial DNA are only exclusively passed down from mother to their offspring, because your egg cell is what has the mitochondria, not just sperms. So that's what passes down mitochondrial DNA. So these mutations can be traced through families as a result. And this is just a picture of mitochondrial inheritance. As you can see, all offspring of the affected individual gets it. And yeah, especially with, like, if it's the mother, all offspring inherits the gene. And then you have multifactorial inheritance, which essentially describes how multiple genes alongside environment can lead to a phenotype. And some diseases you inherited are more genetic, and some diseases can be a mix of genetic and environmental. So that could be something like type 2 diabetes. And then there are some that are completely environmental, and that could be just like communicable diseases or infections. And yeah, and your risk of inheriting something is higher if it's a first degree relative, because you're more likely to inherit those genes rather than a distant relative. So that's inheritance. If anyone has any questions, feel free to put in the chat. Or if not, we'll just move on to questions. So question 1, what type of inheritance does this pedigree show? And just to set the key, the circles are female and the squares are male, and the black squares, so black squares and black circles are affected male and affected female, respectively. Okay, so there's just a mix of 1 and 2, and it is 2 actually. So the reason why it's not 1 is mainly because if you can see here, even though these two parents, it seems like they're not affected. So okay, let me explain better. So it's autosomal because it's like from the pedigree, you can see there's an equal number of males and females are being affected. It can't be X-linked. It's not mitochondrial because if it is mitochondrial, these two individuals would also be getting the gene. And the reason why it's not dominant and recessive is because in this area, you can see that these two individuals are not affected, but they have a child which is affected, which means that these two are likely to be carriers. And carriers are only formed in autosomal recessive inheritance. So that explains why it's not autosomal dominant. So question 2, what pedigree does this type of inheritance show? And again, the same key applies. Squares are male, circles are female. So yeah, I'm getting a few answers for 1, and yes, it is 1. It is X-linked recessive. And the reason why is because it seems from pedigree only males are being affected, and very few of them are being affected. As you can see, there's carriers again in these two, which means that it's X-linked recessive. Question 3, what does this inheritance pedigree show? So I'm getting a few answers, and yes, it is mitochondrial. So as you can see, I got I got one answer for X-linked recessive with, no, sorry, for X-linked dominant. It's not X-linked dominant because even though as you can see, both a lot of the males and the females, there's an equal amount being affected. But the reason why it's mitochondrial is because the affected female, all her offsprings are affected. And in this area too, this affected female, both for children or her offspring are affected, which is very classic of mitochondrial inheritance. So I believe that's all the questions for patterns of inheritance. So now we're just going to move on to genetic mutations and clinical syndromes. Again, if anyone has any questions, feel free to put in the chat, or we'll just move on to genetic mutations. So a mutation is an alteration of nucleotide sequence of the genome of an organism, and this can be a result of DNA replication, mitosis, meiosis, or other types of damage to DNA. Mutations that happen in somatic cells cannot be passed down to the next generation, but mutations that happen in your germline cells can be passed down. So, and there's 3 phenotypic effects. So you have silent mutations, which is genetic mutations that occur but have no effect on the protein formation. And then you have loss of functions, which mean a mutation causes an altered, a loss of function of the protein, is generally recessive as well. And you have gain of function, which is when a mutation occurs and this protein gets a different function from what it's supposed to be having. So there's the 4 main types of mutation that you guys need to know is silent, missense, nonsense, and frame shift. So according to the diagram, I'll just explain it. So this is your normal DNA strand. A silent mutation is when, so as you can see, from the normal, this A has been mispaired and there's a G, but it codes for the same protein, which means that in the end, it's the same protein that's being made. So it's a silent mutation. There's nothing, there's no change in function or nothing, and it's not going to cause any sort of problems. And you have a missense mutation next. As you can see, this mutation causes a different protein to be replaced to the correct protein. And this can cause, typically, maybe, this can sometimes just cause a different this can cause a different structural problem or different functional problem, but it's not really that extreme. And then you have your nonsense mutation, which causes a stop codon to be put. And once you put a stop codon, the next part of your DNA, your proteins are not going to be formed. So this causes a completely dysfunctional protein that has no function, essentially. And then in your frame shifts can be insertions, deletions, and duplications. And what this causes is if you have an addition or a deletion of a base pair, this causes your full reading frame to be shifted because as you remember, it's read from 3 codons. It's read as triplets as 3 codons. So when you remove one, the whole frame shift is moved by 1. So this causes different proteins to be put and makes a very, very different protein from what needs to be made. So that's your gene mutations. Now you have your chromosomal mutations. So there's 2 types of chromosome mutations. You have numerical abnormalities, so you have a different number of chromosomes, and you have structural abnormalities. So the structure of the chromosome in itself is very different. And this can happen, and yeah. So during metaphase of cell division, your chromosomes are most condensed, and hence you're able to karyotype them. And when you karyotype them, you can see that you may be able to see the numerical abnormalities. On your chromosomes, you have light bands and dark bands. So your light bands are euchromatin, which is less condensed, and these are undergoing transcription. As you remember in transcription, it needs to be less condensed for your DNA to be read. And then you have dark bands, which are your heterochromatin, which is more condensed, and those aren't undergoing transcription. So description of a karyotype is your number of chromosomes plus your sex chromosomes and any abnormalities that's observed. So in a typical Down syndrome, you can see Down syndrome is an addition of an extra chromosome. So it's 47XY plus 21, which is a male with Down syndrome. Okay. And then you have your numerical abnormalities. So your numerical abnormalities, one term is aneuploidy. So aneuploidy is just the presence of an abnormal number of chromosomes in a cell. And this can be monosomy, trisomy, or tetrasomy. So monosomy is when an organism has only one copy of a chromosome that should be present in 2 copies. A trisomy is when an organism has a third copy of a chromosome that should be present in 2 copies. And a tetrasomy is when an organism gains 2 extra chromosomes. So examples, the typical examples that we learn are trisomy 13, which is Patau syndrome, trisomy 18, which is Edwards syndrome, and trisomy 21, which is classic Down syndrome. And these abnormalities can occur because of non-disjunction. And non-disjunction is just a failure of your sister chromatids or homologous pairs of your chromosomes to separate. And this can happen in meiosis 1 or meiosis 2. And I just put in these 2 diagrams. As you can see, in this one, the 2 homologous pairs fail to separate. So you get 3 pairs in one, and you get 1 here. So you get 2 with an n+1 and 2 egg cells with n -1. And in this one, in meiosis 2, this homologous pairs could separate it, but the sister chromatids didn't separate. So you get, in one area, 2 normal cells, and in this one, you get n+1 and n -1. And then non-disjunction can also happen in mitosis, and this causes mosaicism, which is that different cells, some cells have the normal amount, while normal amount of chromosomes, and some cells have an abnormal amount, and not every cell is affected. And these are your sex chromosome aneuploidies. The 2 ones that you need to know are Klinefelter's and Turner syndrome. So sex chromosome aneuploidies are a group of conditions in which individuals have an abnormal number of sex chromosomes. So your Klinefelter's is when you have an extra X chromosome. It can be either maternal or paternal. And the name is XXY. And this causes a load of conditions. It causes infertility, gynecomastia, testicular atrophy, reduced facial hair, and feminine fat distribution. And this more in males. And this treatment, the treatment is testosterone from puberty. Then you have Turner syndrome, is when you only have 1 X chromosome. And the newborn diagnosis is when you see redundant neck skin and peripheral lymphedema. You don't really need to know this, it's just extra information, or you just need to know is the aneuploidy, the aneuploidy itself. So what is Klinefelter's and what's the chromosomal abnormality, what's Turner syndrome, and what's the chromosomal abnormality. Chromosome structure, I'm just going to go through this very briefly. You have 4 different types of chromosome structure. So you have your metacentric, where the centromere is in the middle. You have your submetacentric, where it is sort of, it's exactly not in the middle, but yeah, it's just slightly above metacentric. You have acrocentric, which is very much, where the centromere is positioned to give a very short P arm and a long Q arm. And you have your telocentric, where your centromeres are at the terminal end, or your telomeres essentially. And so yeah, there's many structural abnormalities in chromosomal abnormalities. So you have deletions, where a portion of a chromosome is just missing or deleted. You have a duplication, where a portion of a chromosome is essentially duplicated, or duplicated, resulting in extra genetic information. And then you have translocation, where you have a section of a chromosome that is transferred to another chromosome. Just having a question in the chat. How does the position of centromere affect the chromosome? It doesn't really affect the chromosome, but this is how you identify different chromosomes, essentially. Oh, sorry. Yeah, so it doesn't really affect the chromosome in itself. It's just, these are just different structures of chromosomes that you have. And there's nothing essentially wrong with like a metacentric, submetacentric, or an acrocentric. It's just different chromosomes. If that answers your question, no problem. as well as I was saying, you have your translocations, where a portion of your chromosome is transferred to another chromosome. So you have two, your reciprocal translocation. So if you have two chromosomes, this is one section, this is one section, they're essentially switched. And that's what reciprocal means. They're both getting, they're both getting genetic information, but it's just from each other. And then you have Robertsonian translocation, when entire chromosome, so you have, so an entire chromosome is attached at the centromere of another. And going back to the last question, actually, the thing with like acrocentric and telocentric is that, so these Robertsonian translocations are more likely to happen at acrocentric. And this is because they have these tiny short P arms. And that's why it's more likely. And this can happen in telocentric as well, but we mainly learn that it happens in acrocentric. So that's like another thing to add when it comes to the chromosomal structure. And it, in a way, to go back, it does affect, but it's more likely for these translocations to happen in this acrocentric. And then you have your inversions. So an inversion is when a portion of a chromosome is broken off, it's turned upside down, so essentially inverted and reattached. So genetic material is inverted. There's no loss of genetic function, it's just the sequence is changed. And this is just shown in this diagram, this is a duplication, an inversion, a deletion, insertion, and translocation. And here in a Robertsonian translocation, as you can see, your two short P arms, they can sort of go together, form one chromosome. And this is more likely for your small acrocentric and telocentric chromosomes. So if anyone has any other questions, again, feel free to put in the chat, or we'll just move on to the questions. So which of the following matches the correct chromosomal abnormality to its syndrome? Oh, sorry about that. So yeah, the answer is 5 of the above. Patau syndrome is trisomy 13, Edwards is trisomy 18, Klinefelter's is XXY. If someone has one chromosome and no Y chromosome in the somatic cells, they have. Sorry, that's not supposed to be somatic cells. It's supposed to be germline cells. Just a mistype, I'll go and correct that once this is done. But if someone has X chromosome and no Y chromosome in their germ cells, what syndrome do they have? Oh, which answer is correct? So yeah, the answer is Turner syndrome. So again, remember that they only have one X chromosome. Yeah, so question 3, which type of mutation does the following show? So that's a question. Do you know if non-disjunction occurs more in meiosis 1 or meiosis 2? We don't really know when it's more likely to happen, but it can happen equally in meiosis 1 and meiosis 2. You just need to remember if non-disjunction occurs in meiosis 1 or meiosis 2, how does the end egg cells be affected? But that we really don't know if one is more likely than the other, if that answers your question. Yeah, no problem. And yes, I have a lot to go through, so I think I'll just quickly run through this. So question 3 is silent mutation. And then question 4, which type of mutation does this follow? you guys could quickly put in your answers so I can move on to the next section. Okay, so a lot of people are putting 1, which is technically correct, but it's insertion. As you can see, you're adding 1. So it's insertion plus 1. So remember your friendships are insertion, deletion, duplication. So as friendship is correct, but insertion is more accurate. So yeah. Population genetics, just gonna quickly rush through. Here are some different types of traits. In the course of time, I hope you guys will read this and understand. I'm just gonna move on to this section. So population genetics gives the information of prevalence and frequency of genes and genetic disorders and carriers of alleles. So your phenotypic frequency is a proportion of population that shows a phenotype. And your genotypic frequency is a proportion of population expressing a genotype. And your allele frequency is a proportion of alleles in populations. So how you calculate this is in this equation. And then Hardy-Weinberg law is the main point. So Hardy-Weinberg law states that mating is random in a large population with no disruptive circumstances. This law predicts that genotype and allele frequency will remain constant because they're at equilibrium. So essentially what this means is all your gene pool will stay the same regardless if it's a random, if it's a random mating in large population and there's nothing else that's affecting it. And this is a very model situation that doesn't exist in reality. And these are the factors that do disrupt. So mutations disrupt, migration disrupts, because migration, genes are essentially leaving and coming in, so it changes your gene pool. You have non-random mating, which is your inbreeding and sortative. The previous slide, first one you showed us, isn't on the Google Drive. May you add it if it's possible after presentation? Ah, yes. Just let me know which slide it is. Is it, sorry. Is it, if it's this slide, I'll add it to the Google Slides, sorry. Yeah, so you have, but the factors that disrupt are mutation, migration, non-random mating, which is your inbreeding and assortative mating, and your genetic drift. And genetic drift means you're making a smaller population, essentially. And as you remember in Hardy-Weinberg law, it needs to be a large population. These are the two different equations. So p squared plus 2pq plus q squared equals 1, where p is the frequency of a dominant allele and q is the frequency of a recessive allele. And p plus q is equals 1. These are the two things you need to remember. And let's move on to the questions. So which factor does not affect the Hardy-Weinberg law? So yeah, got an answer. It is random mating. For the next two, these are maths questions, so these might take some time. So if you guys could do this, well, it would be much grateful if you guys just see these questions and do this on your own time. And the answers are there, but since these might take some time to go through, and I still have one section of biotechnology to go through, we'll just move on to this. But hopefully you guys see those two questions and do it on your own time. If you have any questions or if you don't get the answer, just let me know. So biotechnology. So biotechnology, the four main things. So one aspect is recombinant DNA. So recombinant DNA is DNA that's made from it's combined DNA from different organisms. And the main thing you use is plasmids, or these ring DNA from bacteria. And how recombinant DNA is made is you use restriction enzymes, which are cutting enzymes that cut at specific DNA sequences to make your DNA fragments. And you use the same restriction enzymes at the vector, so you make complementary sticky ends. So these sticky ends allow the DNA to combine because of the hydrogen bonding and the complementary base pairing. And then you have lastly your DNA ligase that essentially combines and builds this phosphate backbone to form that recombinant DNA. So here's just a picture. Your restriction enzyme cuts, takes a gene of interest, you cut it here, and then you make your modified DNA. How you identify these, one thing you can do is use a gene for antibiotic resistance. And how you do this is you do the same thing, you put a mark for antibiotic resistance, and you put this into bacteria and you culture them in a petri dish, and you put the antibiotics. And so bacteria that haven't taken up, the plasmids haven't taken up that antibiotic resistance, so hence they die out. So the bacteria that do survive are the bacteria that have taken up this recombinant DNA. And you can culture this bacteria and make large quantities of the protein product you want. So mainly use bacteria and yeast because they have a very fast replication rate, so they make a lot of this product. But sometimes you use mammalian cells if you need post-translation modification or if the protein is large, which is something that you can't do with bacteria and yeast. So PCR, polymerase chain reaction, this is how you make rapidly many copies of DNA. And you have 3 steps. You have the denaturation, which is just separation of your strands. You heat at 95 degrees, which is enough to break the hydrogen bonding between the 2 strands. Then you have your annealing. Your annealing is you reduce the temperature to 55 degrees to allow short complementary RNA primers to start to anneal. And then your polymerization or your extension phase is when you put at 72 degrees and Taq polymerase synthesizes new DNA strands. So the reason why you put at 72 degrees and you don't use your normal DNA polymerase is because Taq polymerase, its optimum temperature is 72. And you need to keep this at the temperature range between 95 and 72 to allow this process to keep on happening. If you use our normal DNA polymerase, which has an optimum temperature of 37, it's not going to be as efficient. So that's why you use Taq polymerase, because it's optimal at a very high temperature. Stem cells. So SCNT is a somatic cell nuclear transfer. And this uses stem cells. So stem cells are unspecialized cells that are capable of renewing themselves through mitosis. And under certain conditions, they can be induced to make organ-specific cells like myocardium, so your heart, muscle cell, liver, neurons. You can use it to make any organ-specific cell. So a stem cell transfer is a way they use this to make new organs. So what they do is they take an embryonic stem cell, which is naturally pluripotent, and they remove that nucleus out. So you remove that nucleus out and you put the recipient's nucleus inside, and then you stimulate it to become a blastocyst. A blastocyst cell then can be differentiated to organ-specific cells. An advantage of this is that it reduces the risk of immune rejection because you're putting the recipient's DNA in it, so it's very unlikely for it to be rejected. And you can reduce the wait times for burdens and transplants. But the big disadvantage is the ethical issues of using embryonic stem cells. This is induced pluripotent cells, which is another quite a similar thing to SCNT, and this bypasses the ethical issues. So what this does is this takes an adult somatic cell, and this can be induced to become a pluripotent cell. And there are 4 gene codes that have been found to convert somatic cells to pluripotent stem cells. But the main disadvantage of this is that it has been shown to cause extra cancer-causing genes and fewer tumor suppressor genes, which means you're at a higher risk of getting cancer. Gel electrophoresis, yeah. So gel electrophoresis is how you separate DNA fragments to find a specific sequence you want. So you have DNA, you put restriction enzymes, and you fragment it. And these different sizes of DNA, these different samples are put into wells in a gel, and a current is supplied. So if you can see, if this is a gel, you insert it in different wells at the gel, and you put a current, positive to negative, and you make an electrogradient. So your DNA fragments are naturally negatively charged because of the phosphate. So they move in the direction of, they move to the positive side, because negative and positive attracts. So smaller fragments, they move in the gel faster because they're smaller, and your larger ones take quite some time. So you apply that current for some time, not for a long time, because if you put it for a long time, all the fragments would go at the end, and then there's no point. So you put it for some time, and then you stop the current, and the DNA is then tagged with fluorescence to identify the different sizes. And then the DNA fragments can be seen as bands under UV light, and this represents a DNA fragment according to size. So this is what the gel electrophoresis looks like. So you can see these different fluorescent bands, and all of these, the faster it has traveled, the smaller it is, essentially. And the last thing is gene editing. This is a very niche, very unlikely they'll ask, but since it's in the syllabus, I will talk about it. So you have this gene editing technology called CRISPR-Cas9, which is a very powerful tool which easily allows you to alter DNA sequences and modify gene function. So I have a diagram. So it represents a Cas9 enzyme and a guide RNA. This guide RNA is complementary to the DNA sequence that you want to identify. So what it does is this guide RNA can move and combine with your DNA, the specific DNA sequence that you want to identify. The Cas9 enzyme, then it makes this complex, and this complex is able to cut that specific DNA sequence you want out. And then once it's out, you can put your own pre-designed DNA and make a different sequence. So this can be used, for example, if you have a gene mutation and you have a specific gene that you want to remove out, you can take that mutated gene out and put the correct gene in, and that takes away that mutation. So there's different issues with CRISPR-Cas9. It's the specificity and the accuracy of gene transfer. It also has shown to have a cancer risk, and it can stimulate the immune response, and we don't know the long-term viability. There's a great video from Osmosis that explains this really perfectly. So you can go on this link and check it out if my explanation is absolute garbage. So yeah, that's the end. If you guys, we can do these few questions because hopefully we have time to do it. Or if there's not enough time.
Yeah, sorry. The recording will probably finish soon, but thank you so much, Senara. And as you guys know, the questions and everything are on her slides, and you can always message her. So thank you guys coming so much for today. We'll start again tomorrow at about 10 a.m. I'll have the recording for today and tomorrow. Both of them will be up on either Wednesday or Thursday. So keep an eye out for that. And thank you guys all for coming, and see you tomorrow.
Thank you guys for listening. Bye.